Well, welcome everyone to the fourth edition of the online NanoNeuro meeting. It is a great pleasure to organize this meeting as every year. So my name is Aitzol García Echarri, and I'm a group leader at the Donosti International Physics Center. And today we will have an amazing lineup of speakers talking today. Some of them are already here. So hello, and thanks a lot for accepting our invitation. And Rafa, maybe you want to say a couple words before we start?
Yes, greetings wherever you are in the world. My name is Rafa Juste. I am the director of the Neurotechnology Center at Columbia University. This center started in 9 years ago as Columbia's response to the US Brain Initiative. And among our activities is organizing meetings. And in fact, we've done already 18 different scientific meetings, initially at Columbia. And since COVID, we moved to this online platform. And we're delighted to co-organize this fourth meeting with the DIPC on NanoNeuro, and also delighted to have the support of the Chen Foundation, TCCI. So maybe just a couple of words to explain what is NanoNeuro and why this is important. So you can define NanoNeuro as the intersection between nanoscience and neuroscience, and both things broadly defined. So nanoscience encompasses areas of physics, of chemistry, engineering, and neuroscience, both encompassing basic research and applied research and clinical work. And the reason nanoscience is important, well, it has to do with all these brain initiatives, not just in the US, but in other parts of the world, as there is a push for the last decade to build new methods for neuroscience. And nanotechnology and nanoscience can contribute a lot of very interesting methods. Nanomaterials are very interesting, physical and chemical properties. And those can be used to spearhead new directions in terms of measuring the activity of the nervous system or modifying the activity of the nervous system. And with that spirit, we've organized these meetings to try to get these two disciplines to talk to each other. So in these meetings, the NanoNeuro meetings, you'll find speakers from different areas. It's a very multidisciplinary area. And very often these speakers never go to the same types of scientific conferences together. So this is an opportunity for us to mix in. So maybe we should say a little bit about the program. I'm going to put a link to the program in the chat. And I saw you want to comment a little bit about what the program is going to be and what are the rules for the speakers.
Yeah, absolutely. So we're gonna have 9 talks, if I'm not mistaken, divided in 2 blocks. The first one starts now, basically, and it will last for 2.5 hours, where we will have 4 speakers and 1 panel discussion. So invited speakers have 25 minutes for their presentations, plus 5 minutes of Q&A. And in the second block, which consists also of 4 talks, we will also have a keynote talk, which will last for 40 minutes with 4 or 5 minutes of questions, and then once again a panel discussion. So Rafa shared the link to the program in the chat, and we're still trying to work out the link for the YouTube channel, but it should be on in any minute. And from my side, that's pretty much it. So Rafa, shall we jump in?
Yes, just maybe to mention the people that are following us, you can ask questions during the meeting. You just type your question in the Q&A button that you should have in your Zoom screen. If you're watching this through YouTube, you can also type your questions in the comments, and we're monitoring the channel. And those questions will we will relay the questions to the speakers. The speakers can also ask questions whenever they want to any other speaker, either by typing it in the chat or raising your hand and asking the question viva voce. And we also want all the speakers to remain until the end of their session, because we're going to have a panel discussion between all the speakers and Aitol and I. And with that, Aitol, maybe you can just get the ball rolling with the first speaker.
Absolutely. So it is our pleasure to introduce to all of you Yinwu Chon. And my apologies if I didn't pronounce your last name well. I think we have the same problem. My last name is also strange. And so Yinwu is a professor of chemistry at the Yonsei University in South Korea. And today he will be talking about high performance magnetogenetics. And so whenever you're ready, you can share your screen and you can start your intervention.
Okay, thank you so much for the introduction. And do you see the screen?
Perfectly.
I hope so. Okay. All right. So thank you so much.
Perfect. Yeah.
Is it all right? So I'm Gino. I belong to. I'm really excited to join this symposium. Is the thankful to Rafa and ISO and my institution. There are two institutions that I belong to. One is the Yonsei University Chemistry Department. The other one is Institute for Basic Science, which is another different institute. I'll tell you a little bit about that. About 10 years ago, Korean government started a new initiative for basic science, and it's called the Institute for Basic Science. And then they promote each center to organize team science for, looking for some specific, but it has to be big scientific questions. And this has to be supported for long term, at least 10 years. It could go, decades and so on. And we have centers across the country. My center is located in Seoul. The university is Yonsei, and it covers life science, chemistry, physics, and interdisciplinary topics. So I'm director of the center. It's called a Center for Nanomedicine at Yonsei University. It was founded about 8 years ago, and we have a small graduate program called Nano Biomedical Engineering, and we have about 10 faculty members, 80 students, and postdocs, and the budget is about $7 million a year. And the university, and we have a new building specific for this center. And then we develop nano materials, nano device, nano systems for biological system. And hopefully this could be useful for healthcare, new innovations. And the university, Yonsei, is located in Seoul. It was founded by American missionaries about 135 years ago. And it started as a medical school. So we have the medical healthcare system is called Severance Hospital. And it is probably the largest healthcare system in South Korea, maybe in Far East. And this the picture shows one of the two main buildings, and it is in fact reside in the same campus as you see the old buildings, which is more into humanities and life science and economics and so on. But we have a huge campus located in Seoul and multiple hospitals across the country, and they cover about 4 million patients a year. So it covers about 10% of Korean population, roughly. So that's the introduction of my incident. And some of my questions, because we are interested in magnetism, and this could be a good question to anyone, any scientist, and what are the interaction between magnetism and biological matters. And we all know that we use nuclear spin of water to somehow image out the biological system. That's MRI, and it is related to nuclear spin relaxation time, and we get the anatomic information, molecular information out of it. And then can we do something quite opposite? That meaning writing. Can we do activation or deactivation? Not beyond reading or imaging it. So then how can we do it? Or if so, then how can you do it? And why do we need the nanoscience? And so those are some of the questions that we have. It has been well known that in nature there are many interesting questions, mysteries, and so on. So and then this has been out there for a long time called the magnetoreception, and people argue that or suggest that migratory animals may use the geomagnetic field for their navigation, and still there are some hypotheses and some proposals and so on. So maybe quantum spin dynamics, or light could be important, or maybe just magnetic field itself, and so on. So there are many interesting proposals, and but none is conclusive and still quite elusive. So, but still, people are talking about magnetic GPS system. Somehow, it looks like it is working. The light and magnetism out there for a long time, and obviously, we all know that optical system is probably one of the best medium tool to look into biological system, from molecules, macromolecules and living system, and people have developed many tools. FRET, we all know that fluorescence, that's very important for single molecule study, and STORMSTED, super high resolution imaging, and optogenetics. So we all know that not only you see biological matters, dynamics, and also fine structure imaging, and also controlling the neurons. But one of the limitation beyond the excitement is the large interference with the light and the matter. So that means that the light cannot really go deep inside, only probably a few millimeters and most, and so on. So what we are interested in, what if we use magnetism? Because magnetism, of course, can go very deep. That means that it will be a new system that can look into deep inside the in vivo system. So we have been working on developing a number. So let's say if you are developing FRET, then can you use something called MNET? We discovered that the molecular interaction can be really probed with a magnetism. So we called it the magnetic resonance energy transfer system. We published a few years ago, and then we also developed contrast agent that they can really go into a few micrometer scale. And then the third one is that I'm going to tell you today. It is about magnetogenetics with utilizing a new nanoparticle system. So the nanomagnetogenetics, and basically, we have a neuron, and we want to activate neurons, and there are ion channels, but some of the ion channels can be activated by real mechanical force. Basically, it's similar to what you open your door of your house, the gate, and so on. So we call it as a magneto, but it is associated with a mechanical force, and the force that you needed is about few piconewtons, typically 2 to 3 piconewtons. and then that force is enough to open the ion channel. But the ion channel has to be mechanosensitive ion channel in this case. So the force that we have in biology can span quite large, from nanonewton, micro to nanonewton, and that's antibody-antigen interaction. And then you can go down to piconewton, that's ion channel gating, and so on. So this piconewton force is very important for mechano gating process. And as you can see, the chemical bond is a bit stronger than that. But people are now found that the force signaling in biology is very important. It is very elusive. There are no good tools to really study in real time and with a quantitative measurement and activation. So this is something that we have been interested in. And of course, people have tried utilizing the magnetic nanoparticle and to activate the mechanosensitive ion channel. And this one particular study was really exciting because it was reported that you can really do magnetic control of a nervous system by using ferritin. Ferritin is a well-known protein that can encapsulate iron oxide, and when you do magnetic pooling, basically you gate the mechanism of ion channel, and then it'll do the rest of the work. But a few years later, there are multiple papers mentioning that it's very difficult to reproduce this work simply. And as a chemist and nanochemist, and it's one of the potential reasons for is that the ferritin can generate iron oxide, but this iron oxide is called a ferrihydride. Basically, it is an anti-ferromagnetic material. So given the condition that they performed, the pulling force is very low. It's below attonewton, and then the potential energy is even orders of magnitude smaller than thermal energy. That means the magnetic spin is disoriented. You cannot really have enough force to for the ion channel. And there are some other comment that just looking into the force scale, this may not be that easy to really activate the ion channel. Still is under debate and discussion and so on. So the point is that maybe we need a bigger force. As you can see, the anti-ferromagnetic material cannot really generate enough force. So we need some other force, and of course it made a force, and then we need the force. That's what I think. Yeah. So this is something that we have developed over the years, and then simply you can make a nanoparticle. The nanoparticle can have a quite weak magnetic coercivity, but you can generate fairly high magnetic moment by simply making a assembled nanostructure. These are the iron oxide nanoparticle. It has a high magnetic coercivity, and you just single crystalline, but you coat it with those nanoparticle on the silica bit, few hundred nanometer, typically 500. Usually we use 200. This is a bit old study, but you have a click. Now you can have a nano particle with a high magnetic moment, and then this is the concept that we have. Let's say we have a long distance magnetic alignment, like a magnetic compass that we have. So we have a nanoparticle, and then you utilize a very weak magnetic field, not non-gradient, just a weak field, and at the same time you need some ion channel, which is only activated by mechanical force, and you need a magnetic setup. A magnetic setup should generate torque, because torque you can deliver long distance simulation. So this is the system that we have developed. It's called the magnetic field generator. So when you have a two magnet system like this, of course you can generate magnetic field, but the distance can be quite short. As you make it a bit longer, separate them out, then you will lose the region of, you will have a region of no magnetic field. So you don't want to have that, and then you can put another, you have another, and then you can connect to the magnetic field. So this is called the Halbach array system. So this is what we have utilized. Here you have a let's say magnet bar has about 1 Tesla magnetic field at the end of it, but this will decay quite rapidly, and then it's some reason usually this is a region that we are utilizing, and here the magnetic field strength should be around 20 millitesla. That's what we are using. And then the field grade should be very small, almost planar, and so that you don't have any magnetic gradient field. So and then you rotate this whole magnetic system such that this nanoparticle can rotate accordingly. So this is the video clip that I'm showing you. The particle has a magnetic particle has a fluorescence tag, and then indeed as you rotate the whole rotating magnetic field generator, you accordingly the particle rotates, and then the force that you can generate is roughly 2.5 piconewton, and we are happy with that number. And this is the neural activation. This is a setup that we have. You can rotate any direction. This is a clockwise, counterclockwise. But when you apply the magnetic field, the rotational magnetic field, the fluorescence signal comes out. That means that calcium goes in, and then you can really see the activation of a neuron. And so, as you can see here, we see about significant and quite significant the fluorescence changing, and then the time and the latency time is roughly 100 millisecond, and so on. And the setup is so simple. So you have a array of magnetic, magnet array of magnets that's essentially generate north and south, and then we have behavioral camera, and then the built-in motors, and then we have a motor controller, and so on. So you could see that here are the magnetic field lines, and then the particle will align in this way, as you can see here. And then I'll tell you later, but this we attach it with antigen and attack interaction. And then you rotate this whole system, and then accordingly the particle will rotate, and then it will generate a torque force. So it's the direct force generation, and it's a bioorthogonal. That means it doesn't interfere any biological or chemical system, and then the kinetics is about 100 millisecond, and you can have a different frequency of pulse generation. You could do 1 second. Typically, we use a 1 hertz system, because that's quite reliable too, and it works very nicely. But you could have a faster simulation process by rotating faster and or slower. So you could do quite fast and then longer. They can go up to minutes and so on. Now, what you need is a mechanosensitive ion channel, and as you know that the piezo system has been discovered about 13 years ago here in science paper, and the piezo system is a remarkable ion channel system. You all know that, Adam Patokyan, he won Nobel Prize, and so I met with him probably 2014. I gave a talk at Scripps Institute, and he and I discussed that maybe this is a good system to test our nanoparticle activation for neurons. So he kindly delivered the gene for the piezo. So we let's say we have a neuron, and then you have to do a viral transcription. It's almost same as optogenetic system. So you have this one is a piezo1 MIG-TAC, the adenovirus, and then you do genetic encoding of piezo1 into neuron. And then here you could see that these are super resolution microscope. You can clearly see that the piezo are all very nicely expressed in neuron, and then you link the and you can see that confocal by confocal microscope you can apply magnetic field. Let me just somehow the internet is not that stable. Let me just.
So one suggestion,
Okay.
you would could you turn your camera off? I think that could enhance. Yeah, that could enhance the bandwidth.
Let me see. Camera off. Somehow.
Perfect. Thank you. That may help.
Oh, okay. So, all right. So let me just go back. Somehow, right. So, so here, let me just tell you what it is. So here you have we call it magnetic torque, because we intend to, even though it is spherical particle, because it works as like a compass. So we by using the torque, you generate force. So we call the magnetic torque, and then it is coated with nanoparticle I just mentioned to you, and then coated, and then the surface is covered with PEG, and then here are the antibodies. This antibody will bind into the MIG-TAG expressed on the surface of the ion channel. Actually, in the lateral position, and it is a residue 897. And then it specifically bind into that A97 MIG-TAG with antibody, and antibody is conjugate to the mTOR. So this is, and then you can see that really these particles, the red dots are the particles that we are talking about, and then we apply the rotating magnetic field, and then they're introduced the calcium ions. So then we tested this into the motor cortex to region, and then this is really the motor function of the paw of the mouse, and here you have mouse, and we injected about 2 microliter, the concentration is 15 milligram per ml, and then the mg station is 0.5 hertz about 30 minutes. So 0.5 hertz is very slow rotation, right? And then you can see that. So here the magnetic field is still off, and then it was injected into right hemisphere M2 region, and then let me see somehow. So somehow the video clip is not working properly. I don't know why. Let me just okay. So I mean somehow it is not working properly. The video, I mean, it is rotating, and then the rotation when it is injected in the right hemisphere M2 region, the rotation speed has been increased 5 times, and then you can do the opposite when you inject it into M2 region of left hemisphere, and then it does. So this tells you that the magnetomechanical simulation is working, and now you could consider realize that this is about activation. Maybe you could go into inhibitory ion channel with the same concept. And then you can also go for deep brain stimulation. Maybe you could do neurodegenerative studies and so on. And hopefully sometime neural circuits concept. But today I'll tell you a little bit about cell specificity, because it has been well known that the neural specific neuron, the cell type selectivity is critical to be a useful tool for neuroscience. So here we are interested in the network hypothalamus, and there are multiple different type of cells, and then we are in specific the we get, and then the technology that utilizes from the very well known Krylov speed system. You all know that this is quite effective in many biological systems, but we want to know whether this technology can be implemented into our magnetogenetic system. So here you have a Cree mouse, and then some of the cells are actually positive, and then now we have a double inverted the open reading frame, the LOX and the piezo1 and LOX, and then the viral delivery only goes into the positive cell lines, so it generates a piezo1. Then you attach the nanoparticle, and then you can clearly have a piezo1 here, and then here is mTOR, and then the image shows that there is very nice coincidence, and then we do mechanization. So the neurons in LH region is quite important, and then one case for V specific activation can relate with feeding and social interaction. So that's what we have done, and here this is we get specific engines in real-time feeding, and this is experiment condition. So you have a free mouse and then free piezo1 viral injection, and then we wait some time to express the ion channel, and then we inject the mTOR and do the MG simulation. And MG simulation is about the same as the one before, 0.5 hertz, and then total 5 minutes. So this is a same mouse. This is a field zone, and this is a magnetic field is off, and so it's just roaming around. The video is somehow doesn't work. I don't know. There is some interference with my PowerPoint presentation and the video or internet broadcasting. I don't know why, but it doesn't work. So, but quite focused into food just roaming around. And so this is a cumulative duration time, and then it is increased about in least 3 times higher. And then the DAPI C4 shows that clearly these neurons are activated by the mechanosensitive ion channel stimulation. So that's quite exciting to us. And then you extended this study into social novelty study with multiple animal studies. So here you have 2 mice, and there is M1, M2, and M2 is a novel, and M1 is familiar. There is a mouse in each cage. And then this is a control experiment, and it's better. They just roam around. There are 2 mice, and then just go to M2 and also M1. This is the mouse, the track is shown, so it goes almost there. But with the magnetic stimulation, the video doesn't really work. So as you can clearly see that they spend most of the time with M2, and this is because of the VGAS stimulation. When you do the VGAS stimulation, they increase the social behavior, and then it go into M2 region. So this is our study with a piezo. We can clearly see that more interaction time into M2 region. And this is a study by optogenetics, and it's comparable. And one clear distinction is that we utilize 2 mice, whereas they use only 1 mice. The mouse was optically, linked into the system. So, but we could say that because our system is a wireless, and also we could long distance simulation. I think that could be quite exciting. So this is the summary of our system. So you have adenovirus PH1, and then M2 nanoparticle, and the ion channels I talked about the piezo one, but you could utilize any mechanism of ion channel, and you could patch to adenovirus lantern AAV. And the particle, what we used was about 200 nanometer to 500, but you can tune the force. If you need 7 piconewton, 10 piconewton, you could do that. You could go down to 2 piconewton and so on. And biocompatibility, and these are non-toxic, non-leaching, meaning no metal ions are leaching, because it is surface is well coated with silica and so on, so it's well protected. And the apparatus is quite easy, and it can have a quite large long distance working area, and you could go into primate and so on in principle, and the channel gating frequency can be tuned. Not as good as optogenetics, because you could have a ultra fast and an ultra slow and medium. And the point is that this is a wireless energy transfer 4th generation. You could do that, and then it's a bioorthogonal and a reliable kinetics. Then you could do all kinds of molecular and cellular specificity with it. So people have done beautiful, amazing studies with optogenetics. And maybe magnetic genetics can be another potential tool for going to maybe more complex or bigger scale object, and that's something that we are interested in. Maybe you can go to multiple targets at once in a large area, going to clinical studies for brain disease. At the same time, we could also go for neuropsychiatry studies and so on. So this could be, maybe complementary. And this slide
Right.
shows that you all know that magnetic tweezers,
Jimbo.
optical tweezers, and so on. Yeah.
Yeah, we are over time by 10 minutes
Oh, is that
already.
right?
So
Okay, so
please,
this
if
is
you can summarize.
our last slide. So just telling you that this working distance is quite important, and we can really show you that force and the distance can be tuned, and we are actually well, this is we have UI controller and Mac 35 and 6 centimeter. We are interested in, actually, we are looking for neuroscientists who can work together to develop our tool, with a magnetic and so on. If you are interested in, please visit the website, and then we could, communicate more. And with that, I'd like to close my talk, and thank you so much for your attention.
All right, thank you so much, Ibu. So we are absolutely over time. So if you don't mind, we will move the questions to the panel that we will celebrate after all the talks in this first session. And so without further ado, I would like to introduce our next speaker, Shigeki Kiyonaka. Once again, my apologies if I didn't pronounce your name well. It's not easy for us. And so Shigeki is an associate professor at the Nagoya University in Japan. And today he will be talking about chemical approaches for understanding physiological roles of glutamate receptors in neurons. So please, if you can share your screen, remember that you have 25 minutes plus 5 minutes of questions. And whenever you want, you can start. Thank you very much.
Okay, thank you for your introductions. Can you hear me? Is it fine? Okay, that's fine.
Perfect.
My name is Shigeki Kiyonaka from Nagoya University, Japan. I would like to thank the organizer, especially Raphael and Arizal, for giving me this great opportunity to present our researches. Our research direction is understanding brain function by chemical approaches. One research direction is visualizing neurotransmitter receptor by chemical labeling technique. Another direction is functional control of the receptor proteins by chemogenetics approach. First, I talk about fluorescent visualization of neurotransmitter receptor by chemical labeling. In excited neurons, glutamate receptors are highly expressed in the postsynaptic side, which are categorized into ion channel types and GPCR types. The ion channel types are further divided into mainly three classes, kainate, NMDA, and AMP receptors. Among them, AMP receptor has essential role in synaptic plasticity, a putative cellular mechanism of our memory. Recent studies have revealed that the number of AMP receptors is strictly controlling during synaptic plasticity. For example, during long-term potentiation, excitosis and lateral diffusion of AMP receptors are facilitated to increase the number of cell surface AMP receptors on the postsynaptic membrane to enhance postsynaptic current. In contrast, in the long-term depression, endocytosis and degradation pathway are accelerated to decrease cell surface AMP receptors. To visualize this event, AMP receptor tagged with pH-sensitive GFP variant on the N-terminus have been widely utilized to visualize cell surface AMP receptor selectively. However, in most cases, as I can show you, GFP-tagged AMP receptors are overexpressed in the neurons. As I mentioned, the strict control of the number of AMP receptors are essential. However, the number of AMP receptors are already changed by overexpression of AMP receptors, GFP-target AMP receptors. Thus, we should visualize endogenous AMP receptors. The potential approach would be utilization of antibody. However, considering the narrow size of the synaptic cleft, antibody cannot visualize synaptic AMP receptors. Thus, ideally, native AMP receptor should be tagged with small fluorophore. To visualize the endogenous amperester, we focus on chemical labeling technique termed ligand-directed acetylimazole LDI chemistry developed by Professor Hamachi, my previous boss. In this method, we utilized labeling reagent having the ligand group and fluorophore which are connected via reactive acetylimazole linker showing moderate reactivity. After addition of the reagent in the culture medium, the ligand group is recognized to the target proteins. As a result, the fluorophore is transferred to the target protein by covalent bond formation with lysine or serine residue. And the resultant ligand group can be removed by simple medium exchange. To apply this method to amperester, we designed labeling reagent for amperester termed CAM2 or G, which has selected ligand to amperesters and organ green as a fluorophore. These are connected via reactive acetylimazole. First, the chemical labeling was examined hex cells transiently expressed with amperesters. We need several hours for the chemical labeling because of the moderate reactivity of the CAM2 reagent. Thus, the cells are needed to keep at low temperature such as 70 degrees during the labeling period to suppress the internalization of the labeled amperesters. This confocal image was obtained after adding the labeling reagent to the culture medium. This indicates that labeling reagent does not penetrate into the cells. And the cells are incubated 3 hours at 70 degrees. After the medium exchange, confocal image clearly indicates a strong fluorescent signal from the cell surface. To confirm the selectivity to amperesters of the labeling, we examined Western blotting using anti-organ green antibody after the chemical labeling. And zinc labeled band was observed corresponding to the molecular weight of amperesters. Importantly, the labeled band disappeared in the presence of competitive inhibitor. Indicates that selective labeling to cell surface amperester under live cell conditions. Then we applied this technique to label amperester endogenously expressed in neurons. In these cases, primary cultural neurons were prepared from cerebral cortex. As is the case of the labeling in hex cells, culture reagent was added to the neuron and incubated 4 hours at 70 degrees. I show here the Western blotting using anti-organ green antibody indicates selective labeling to amperester endogenously expressed in neuron with fluorophore. Next, we examined fluorescent visualization of endogenous amperester in the cultured neuron. In this experiment, cultured hippocampal neuron were incubated with CAM2 AX488 having RX488 instead of the organ and the live cell condition. To visualize the shape of neurons, after fixation with paraformaldehyde, dendrites were visualized using anti-MAP2 antibody. As shown here, the green labeled signals were observed along dendrites, as shown here. Importantly, the fluorescent labeled puncture merged well with the anti-PSC95 antibody, indicates that we succeeded in chemical labeling of endogenous amperester highly expressed in the synaptic puncture in neurons. We succeeded in chemical labeling of endogenous amperester by LDI chemistries. However, we have some limitation in the original method. First, this experiment should be conducted at 70 degrees to suppress endocytosis of amperester during the labeling period. Moreover, we need to incubate the neurons with serum-free medium during the labeling. This is because hydrophobic and aromatic fluorophore is recognized to albumin included in the serum, which causes non-specific labeling. To improve this drawback, we developed a modified method by combining the LI chemistry with biosonal chemistries. In this method, tissue group click chemistry donor is covalently labeled to unprecedented. We can conduct chemical labeling even in the serum containing medium at 37 degrees because TCO is relatively small hydrophobic group compared with large fluorophore of the original method. After tissue labeling, fluorophore is covalently attached to unprecedented using TCO and tetragen ligation, one of the fastest quick reaction. Although the labeled unprecedented are internalized during the first step, cell surface unprecedented are selectively visualized in the second step. Okay. First, we examined the two-step labeling in hex cells transiently expressed unprecedented. In this experiment, CAMS tissue having tissue group was incubated at 37 degrees. In this experiment, CAMS tissue group was incubated at 37 degrees and added tetragen probe for 5 minutes. The labeling was analyzed by Western blotting, as shown here. In the CAMS tissue labeling, unprecedented was selectively labeled even in the presence of serum. Importantly, the label in the original method at 17 degrees, in the original method means direct labeling method. Confocal imaging, this image shows that the labeled unprecedented were internalized as shown here. In contrast, in the current two-step labeling method, labeled unprecedented are selectively visualized on the cell surface. Another advantage of the two-step labeling method is that tetragen and fluorophore show high fluorescence after tetragen tissue ligation. Tetragen group act as a fluorescent quencher, and this quenching effect is significantly weakened after reaction of tetragen and TCO. Thanks to the turn-on type fluorescent properties, we can estimate the reaction kinetics of the second fluorophore labeling on the cell surface by adding the tetragen probe in the culture medium, which determined fluorophore labeling was finished within 3 minutes. Then we applied this method for visualizing cell surface unprecedented in neurons. In this experiment, CAMS tissue was incubated for hours at 37 degrees. Confocal imaging after immunostaining clearly indicates that endogenous unprecedented were selectively labeled in neurons. In this case, tetragen Alexa 488 was added for 5 minutes. Another advantage of the two-step labeling is that we can easily exchange the labeled fluorophore in this method, and we label theta 647 using tetragen theta 647 instead of the tetragen Alexa 488. This probe, theta 647, is highly stable and show unique fluorescent lifetime. Thus, using the fluorescent lifetime imaging microscopy, we can extract theta 647 component from the fluorescent image, which allow quantification of cell surface unprecedented by subtracting the background signals. This reveals that the ratio of unprecedented in dendrite and spine was 3:10. Next, we analyze the lifetime of endogenous unprecedented in neurons using two-step labeling method with biochemical analysis. In this experiment, the cortical neuron were incubated with CAMS tissue for 10 hours at 37 degrees, and the neuron were incubated for each period, such as 0 to 4 to 120 hours. After the incubation, the remained surface unprecedented were labeled with tetragen FL. Then analyze FL labeled unprecedented by Western blotting. I show here the half-life of cell surface unprecedented was 33 hours in neurons, okay? We also determine the half-life of AMPA receptor in HEC2 CT cells in the similar method. Interesting, the value was significantly shorter than that obtained in neurons. This difference was curious for us. Then we analyze the difference in neuron and HEC cells by focusing the recycling of AMPA receptors. To analyze the recycling AMPA receptors, after come to tissue labeling, 4 hours in neurons, surface AMPA receptor were labeled with tetragen FL as a first pulse, okay? And incubated for 15 minutes. Then the tetragen RX647 was incubated as a second pulse to label exercise dosed. In other words, recycled AMPA receptors. Western blotting clearly indicates that the recycling AMPA receptors were observed in cultured neurons. However, any recycled AMPA receptors were not observed in HEC cells. In summary, our two-step labeling reveals the lifetime of AMPA receptor is significantly different between neuron and HEC cells. And one of the reasons is that the HEC cell lacks the recycling process of AMPA receptors. As a next directions, we are now analyzing recycling process of AMPA receptor in more detail by using the two-step method, by modifying this method. In this case, after surface labeling, the cells were fixed, okay, surface labeling after fixed. Then they performed the second step labeling targeted for intercellular component. As shown here, the intercellular AMPA receptor was selectively visualized as a different color in HEC cells. And some component after internalized has been visualized with a different color. So we think that this method would be used to analyze the recycling process of AMPA receptor in the future. Okay, I would like to move another topic, chemogenetics of cell surface receptors. One of the frontier of neuroscience is understanding the neural network, as you know. This new direction has been achieved by developing technique for activation of target cells, such as optogenetics and chemogenetics, maybe another magnetogenetics. Here we focus on chemogenetic approaches. The representative is DRET technique. In this technique, muscarinic acetylcholine receptor is mutated to response to the designed ligand CNO. This receptor lacks affinity to the endogenous ligand acetylcholine. Thus, expression of the designer receptor in the target cell allows conducting downstream G protein signals by designed ligand. One of the advantages of the chemogenetics is controlling the cellular function even in the deep region of brain. Actually, after expression of DRET protein using adenovirus virus in the target cells, oral administration of CNO controls the target G protein signal in a cell type specific manner in non-invasive fashion, even in large animals. DRET technique is undoubtedly powerful for controlling nerve activity by chemicals. However, DRET protein lack original ligand binding, and the DRET protein are overexpressed in most cases. Thus, the ligand-induced signals are largely different from that induced by endogenous ligand receptor interaction. Thus, it is difficult to apply this method for analyzing the physiological role of the endogenous receptors. We think that the next direction of chemogenetics would be molecular target to chemogenetics, in which the receptor of endogenous receptor should be controlled in a cell type specific manner, like here. In this direction, 2 points are required. First is selective activation of target receptor by designed ligand by introducing mutation in the target protein in a cell type specific manner. Another important point is original receptor function should not be affected by the mutation. Here we focus on metabolic glutamate receptors mGlu1 as the target proteins which belong to class C GPCRs. mGlu1 is highly expressed in the cerebrum, which has essential role for motor learning. mGlu1 has composed of extracellular ligand-bind domain and transferrin regions. Structural analysis reveals that the glutamate binding to the ligand-bind domain induced domain closure of the ligand domain and the structure change, this structure change transmitted to the transferrin region for activation of the receptors. We focus on the structure change. And our strategy is introduction of histidine mutation on both upper and lower lips of the domain. And metal coordination to the mutated histidine residue would induce domain closure. We expected that if the structure change are comparable to that induced by glutamate binding, the metal coordination can activate the mutant receptor selectively. Of note, this mutation site does not join the glutamate binding. Thus, original receptor function would not be affected by the mutations. Based on the structure information of the ligand-bind domain, mutation site was selected where distance between upper lips and lower lips become close after glutamate binding. We designed this double cysteine mutant, and functional assay was examined by calcium imaging. We first examined glutamate-induced response in each mutant, and original cell function are completely retained in the 5 mutant. Next, we examined effect of metal ion or metal complex for chemogenetic expression of these mutant. RAF screening identifies the metal complex, paradigm complex, which activates 3 of the mutant. Among them, we selected a mutant. It is highlighted. As shown here, after addition of the paradigm complex in the culture medium, prominent calcium response was observed in the cells transfected with the W mutant, but not with the wild-type mGlu1. In our strategy, histidine residue are required on both upper and lower lip, as I mentioned. To confirm our concept, we prepared single mutant, okay? And we expect that this mutant failed to be activated by paradigm complex. As shown here, the A59H mutant failed to be activated as we expected. However, N264H mutant was activated by paradigm complex, although the mutant lacked histidine residue on the upper side. We searched other coordinating histidine on the upper side, and we revealed that point mutagenesis reveals that the endogenous H55 coordinate paradigm complex. In other words, point mutagenesis of the N264H is sufficient for chemogenetic activation. Those dependency of paradigm complex clearly indicates that the single mutant was selectively activated by micromolar of paradigm complex. Importantly, glutamate-induced response were not affected by the mutation as we expected. So glutamate response is completely kept even in the mutant. Now we obtained the glutamate mutant and ligand pair, paradigm complex, or chemogenetic activation. Prior to apply this method to neurons, we examined toxicity of the metal complex to neuron-like PC12 cells. As shown here, the neurite outgrowth of PC12 cells were not affected by the paradigm complex. However, the cell growth was suppressed by high concentration of paradigm complex. In fact, we speculated this possibility, cell toxicity, because the chemical structure of paradigm complex looks like cisplatin. So the inhibition of cell growth would be suppressed by DNA binding of this complex. In other words, we expect that the decrease of the cell permeability of the metal complex improves the cell toxicity. Then we designed 4 hydrophilic pyruvate complex having hydroxy or sulfonate group to inhibit the penetration of the cells of the complex. Among them, more substituted complex successfully activates the mGlu1 mutant. Regarding the cell toxicity, sulfonated version of the complex termed pyrazine sulfo-BP does not affect the cell toxicity even in the high concentration. Thus, we utilize pyrazine sulfo-BP for chemogenetic activation of mGlu1 in neurons. To chemogenetic activation of mGlu1, we prepared the knock-in mouse having N6OH mutation by CRISPR-Cas9 technique. In the mutant, not only expression level of mGlu1, but also the function of mGlu1 were comparable to those of wild-type mice. Then we examined chemogenetic activation of mGlu1 in neurons. Here we focus on cerebral LTD. Previous study revealed that activation of mGlu1 in Purkinje cells by DHPG, a selective ligand, induced sustained decrease of EPSP, which is mediated by calcium influx via AMPA receptor, which is known as chemical LTD. Then we applied pyrazine sulfo-BP in the current chronic mode in Purkinje cells in acutely prepared cerebral slice, which successfully induced sustained decrease of EPSC. This indicates that our system can be applied for chemogenetic activation of mGlu1 endogenously expressed in neurons. We also succeeded to apply this method for cell type specific activation of mGlu1 using AV in cerebral slices. Please see our paper for the details. Okay, one of the characteristic features of our method is this method can be applied to other receptor family showing the same structure change, such as classic GPCRs, other GPCRs, and ion channel type glutamate receptors. So the current method would be one of the useful tools for molecular targeted chemogenetics. In my talk, I reported recent progress for chemical labeling and chemogenetic of glutamate receptors. Now we are developing other method which can be applied in vivo for understanding physiological role of glutamate receptor and other receptor in not only neuron but also in non-neuronal cells, glial cells. Finally, I would like to thank many collaborators, especially for my previous boss, Professor Hamachi, and current laboratory member, and other collaborators such as Professor Yuzaki and Kakigawa in Keio University for electrophysiological assay in neurons. And I also thank Dr. Takahashi and Mizuno in Tsukuba University for making knock-in mouse. Thank you for your kind attention.
Now we have a couple minutes for questions. We're running late because of the delays that we had at the beginning with technical issues, but if there is any question either from the audience or the rest of the panelists, and if not, we move the questions, the Q&A to the panelists, to the panel at the end. But Rafa, you have a question, so go ahead.
Yes, this was exquisite, Shigeki. Congratulations.
Yes, thank you very much.
For the chemogenetics part, why do you need the metal? Because it's probably the bipyridines that are doing the binding. No? So couldn't you just do it with an organic compound without any metal coordination? I don't understand why the metal is critical here.
Okay. In the metal coordination, we can control the angle of the binding. Okay, this is good for fix the conformation of the protein structures. Especially, can I share my slide?
Yeah.
Okay, and pyrazine bipyridine complex is very useful because two coordination site is already fixed with this ligand. So only two histidine can bind to the pyruvate complex, and this angle can be fixed to 120 degrees. So we can manipulate the protein structure precisely by using this complex.
So it's more rigid. It's more rigid, the angle, and then you change the rest.
Yeah, in the small molecule, it is surely another possibility is that covalent bond formation by small molecules. However, in this case, the bond is non-covalent. Irreversible direction. However, the metal in the case of metal coordination, this is a reversible bond formation. So we can wash out the metal complex after medium exchange. This step is reversible. So we can control the activity.
Excellent. So if you don't mind, we could pass the word to Xiaoji Duan. He is an associate, well, first of all, thank you so much for this excellent talk and for answering the questions, Higeji. And so I was introducing Xiaoji Duan. Xiaoji is an associate professor in the College of Future Technology in Peking University in China. And today she will be talking about nano-enabled brain-wide neural interface, which is a dream to us. So I'm very, very excited and very looking forward to hear what you have to tell us. So without further ado, the conference is yours. Thank you. You are muted, sorry.
All right, okay, sorry.
Now.
Thank you for the introduction. My name is Xiaojie Duan from Department of Biomedical Engineering, College of Future Technology at Peking University. It's a great honor to be here and share some of our recent progress in the field of nano-enabled brain-wide neural interfacing. Okay, sorry. So we know that most of our behaviors are not simply the result of activity in small sections of the brain, but rather the coordinated efforts of neural activity across a brain-wide scale. So let's take the visual perception as an example. When we see something, information from our retinas gets sent to the primary visual cortex. There it is combined with inputs from other brain regions responsible for object recognition, motion detection, and visual attention. It's this synchronized and coordinated activity across these different networks that allows us to make sense of what we are looking at. From this perspective, in order to understand the neural processes that drive behavior, it's important to study the activity of the brain on a brain-wide scale. So we have several techniques to map neural activity across brain. Some notable methods include an electroencephalogram, EEG, an MEG, that is magnetoencephalography, and functional magnetic resonance imaging and functional near-infrared spectroscopy. These techniques allow us to observe and record brain activity across a wide spatial scale without invasive procedures. However, they do have their limitations when it comes to spatial temporal resolution and signal bandwidth. Additionally, it is worth noting that fMRI and fNIR rely on blood oxygenation level dependent, that is both signals, through neurovascular coupling, which indirectly reflect neural activity. So in order to explore alternative techniques for recording neural activity throughout the brain, our team has been focusing on two major directions. First, we are utilizing advanced microfabrication methods and incorporating nanomaterials to develop large-scale implantable electrode arrays or probes. This innovative approach is coupled with our commitment to minimizing the invasiveness typically associated with such electrode arrays. This approach allows us to achieve extensive coverage with high spatial temporal resolution with recording brain activity. And the second, we are working on combining different techniques such as electrophysiological recording or stimulation with magnetic resonance imaging. This integrative approach capitalizes on MRI's remarkable ability for whole brain structural and functional mapping. By bringing these techniques together, we are expanding the application possibilities of MRI for studying the intricacies of the brain. So now, let's delve into the first direction we are working on. Our focus here lies on intracranial electrocorticograms techniques. ECOGs allow for the direct measurement of neuroelectrical activity by employing a planar electrode array placed on the surface of the cortex. By utilizing a large area electrode array, we can easily expand the coverage of our recordings. The proximity between the signal source and the detector in ECOGs results in outstanding signal quality, achieving remarkable spatial temporal resolution as micrometer and millisecond respectively. Additionally, ECOGs have a very high bandwidth up to 500 Hz. However, it is important to note that ECOG recordings need a craniotomy, and the subdural recordings require an additional durotomy to expose the cerebral surface of the brain. These surgical procedures involved, particularly for large area ECOG arrays, bring forth various operative and post-operative risks, including infections, inflammatory responses, as well as the potential for brain hematoma or edema. Consequently, the invasiveness and associated surgical risks present considerable challenges when it comes to implanting large area ECOG arrays for recording brain-wide neural activity. In addition, apart from the surgical risks, the long-term implantation of ECOG arrays on the brain surface gives rise to additional concerns. You can see here, there are actually intricate connections and crucial mass exchange occurring between the skull and the dura mater, as well as between the dura mater and the brain surface. So when a large area ECOG array is chronically present, it can disrupt these vital connections, hindering the proper flow of necessary substances. As a result, complications like inflammation and brain edema can arise, posing further challenges to consider. So in order to overcome these challenges associated with surgery risks and ensure long-term biocompatibility for large-scale ECOG recording, we drew inspiration from the application of the self-expandable nitinol stent and developed a unique device called the shape-changing electrode array, or C for short. This device consists of a thin MEA film, only a few micrometer thick, which is coupled with a shape actuator. Before implantation, the C is deliberately deformed into a narrow strip, measuring just a few millimeters in width. This compact form allows for easy insertion into the cortical surface through a very small opening in the skull for epidural recording, or a small slit in the dura mater for subdural recording. Once inserted, the shape actuator comes into play, facilitating the shape transformation of the C. It unfolds and reshapes itself into an ultra-thin sheet. After retracting the shape actuator, a large area conformal interface can form with the cortical surface, thus enabling brain-wide activity mapping with high spatial resolution and bandwidth. We employed several strategies in the design of the C to address the unique challenges and requirements. Firstly, the electrode array incorporates a multi-layer structure utilizing a web-like carbon nanotube conducting film as its conducting material. This is a vital component of the C, as it allows the electrode to maintain its functionality even during the shape transformation process, which involves very extreme strains. The web-like thin film is composed of interconnected thin bundles of single-walled carbon nanotubes, ensuring conductivity even under substantial mechanical deformations. To ensure mechanical compliance and a conformal interface with the brain surface, the array was fabricated on a thin parylene film measuring only 4 micrometers in thickness. Secondly, the shape transformation is facilitated by a nitinol actuator. As a shape memory alloy, nitinol exhibits stable austenite and martensite structures at different temperatures. By heating the nitinol beyond its transition temperatures, it undergoes a transformation from the martensite structure to the austenite structure, ultimately returning its original shape and size. Lastly, we integrated a mesh structure within the electrode array film. This design serves two important purposes: ensuring proper connection formation and facilitating mass exchange between the skull and dura mater, or between the dura mater and the brain. Through our research, we discovered that this mesh structure is very critical in maintaining a high chronic biocompatibility of the large area ECOG arrays. Using these strategies, we successfully implanted the electrode arrays, which covered nearly the entire hemisphere of the brains of the rat into the epidural space of adult rats through a very small cranial opening, measuring only 0.8 millimeter in width and 2 millimeter in length. Here is a video showing an implantation process of a C into a rat. In this particular case, we thinned the rat's skull to provide better visibility during the deployment of the C. Throughout the entire implantation process, we observed no bleeding. Once we removed the deployed C from the rat's brain, we discovered that the brain region underneath the electrode array remained completely intact. There was no visible damage observed on the brain tissue or the blood vessels, as illustrated here. This serves as evidence that the implantation of the Cs did not cause any acute damage to the brain. The strategy of utilizing Cs for minimally invasive intracranial brain activity mapping can also be extended to large animal models. In our work, to minimize the signal attenuation caused by the thick dura mater, Cs were implanted subdurally in the large animal application. The image displayed here captures the deployment of a C in the subdural space of a beagle brain. The C used in this case measured 20 millimeter by 15 millimeter in size. It was inserted into the subdural space through a dural slit about 6 millimeter in length without removing any part of the dura mater. Similar to our previous findings, we observed no bleeding during the entire implantation process. Both in the rat and the beagle cases, the Cs adhered tightly to the brain surface, forming a very conformal interface. Crucially, after the Cs underwent a round of shape transformation, only a very small fraction of channels exhibited a slight increase in impedance, and the majority of channels remained within the required range for successful neural recording, indicating the preservation of their functionality throughout the shape transformation process. To assess the impact of the C implantation process on the brain, we conducted MRI studies. The T2 structural scans revealed no discernible abnormalities in the brain's anatomical features or shape following the implantation. Additionally, the T2-weighted dynamic contrast-enhanced MRI showed no noticeable irritation to the blood vessels in the dura mater or brain resulting from the implantation process. Furthermore, there's no time to explain the details, but the histology studies show that in the acute stage, the C implants elicit a very slight inflammatory response. However, this inflammatory response diminished quickly over time. These results confirm both the minimally invasive nature of the C implantation process and also the excellent long-term biocompatibility of Cs. With Cs implanted through this minimally invasive procedure, we mapped high-quality microECOG activities from rodent brains during seizures and also from canine brains during the emergence period. Significantly, the Cs not only recorded the low-frequency components accessible through non-invasive techniques, but also captured high-quality, high-frequency oscillations reaching frequencies of up to 500 Hertz. These recordings revealed the spatial temporal organization of different brain states with high resolution and bandwidth that cannot be achieved using existing non-invasive techniques. Next, I will proceed with the second part of my presentation, which focuses on the development of MRI-compatible electrodes for simultaneous deeper brain stimulation and fMRI studies. DBS has become a widely used treatment for movement disorders like Parkinson's disease, which also showing promise as a strategy for managing treatment-resistant depression (TRD). Despite its proven efficacy, the underlying mechanism of DBS remain elusive. Functional MRI represents as a powerful tool for mapping brain activity on a whole brain scale. By combining DBS with fMRI in a simultaneous manner, we can gain invaluable insights into the modulatory effects and the therapeutic mechanism of DBS in a wide range of neurological disorders. This exciting avenue could bring us closer to unlocking the full potential of DBS. One major hurdle in DBS fMR studies stems from the considerable image artifact produced by traditional materials used for DBS electrodes during MRI scanning. As an example shown here, a platinum iridium electrode measuring 100 micrometers can generate artifact several millimeters in size. These artifacts create obstacles when attempting to image the brain regions surrounding the electrodes. Consequently, obtaining a complete and unbiased activation pattern of DBS becomes unattainable. We explored the use of graphene for fabricating DBS electrodes. In contrast to platinum, graphite exhibits a significantly closer magnetic susceptibility to water. Our approach involved fabricating electrodes using graphene fibers, which consists of densely stacked and aligned graphene oxide sheets running parallel to the fiber's main axis. These graphene fibers possess high porosity and roughness, resulting in very good charge injection capabilities. In fact, their charge injection capacity surpasses that of most commonly used materials for neurostimulation and is only slightly lower than that of PDOT. Moreover, our tests have demonstrated that graphene fiber electrodes exhibit superior stability when compared to PDOT electrodes. Importantly, the graphene fiber electrodes showed excellent MRI compatibility. When subjected to a 9.4 Tesla MRI, these electrodes produced an artifact in T2 sequence that was 8 times smaller than that generated by platinum iridium electrodes. Furthermore, in fact, in EPI sequences, the artifact was 4 times smaller comparable to platinum iridium electrodes in terms of MRI artifact reduction. In fact, not only did the graphene fiber electrodes outperform platinum iridium electrodes, but they also demonstrated smaller artifacts compared to various other electrode materials, including tungsten and carbon fiber. We utilize the MRI-compatible graphene fiber electrodes to investigate the DBS effects targeting the lateral habenule LHB in depressive rat models. The LHB, known as a negative reward center, has shown potential as a valuable DBS target in treating TRD. However, LHB has a very small size, measuring only 3 by 3 by 3 millimeters in the human brain. This compact size presents significant challenges for study with DBS electrodes in human subjects. We implanted graphene fiber electrodes into the LHB of 2 depressive rat models with Kyoto WKY rats, which represents endogenous depression, and rats treated with lipopolysaccharide LPS. These rat models exhibit depressive-like behaviors, including reduced preference for sucrose, increased immobility time in forced swimming test, and decreased locomotor activity in open field test, indicating anhedonia, despair-like behavior, and anxiety-related symptoms, respectively. When we examine the LHB using MRI, we observe the minimal artifact induced by the graphene fiber electrodes, allowing us to easily and accurately verify the correct placement of the implanted electrodes within the LHB. Remarkably, when we deliver the high-frequency stimulation into the LHB using the graphene fiber electrodes, these depressive rats experience an immediate antidepressant effect. This was evident through increased locomotor activity observed in the open field test, the reduced immobility time in the forced swimming test, and also a significant rise in sucrose preference. It is important to note that both the WKY and the LPS treated depressive rats responded positively to LHB DBS, demonstrating a high success rate for this stimulation technique. To gain further insight into the mechanism behind this immediate antidepressant effects of LHB DBS, we conducted fMRI scans on the same groups of WKY and LPS treated rats that was subjected to the behavioral tests. By analyzing the correlation between the antidepressant response and the fMRI signals from various brain regions, we aimed to shed light on the underlying mechanism. We observed the robust positive bold responses ipsilaterally along both the direct afferent and the efferent circuitry of LHB. These regions include the medial prefrontal cortex, MPFC, dorsal-raphe nucleus, DRN, ventral tegmental area, VTA, and some other areas. And also we found the activation in some other areas, which is beyond the direct circuitry, such as the most of the activity area are in the direct connections with LHB, but there are 3 regions which are not in the direct circuitry. That is SLEA, cingulate cortex, and retrosplenial cortex, RS. We performed the Pearson correlation analysis to examine the relationship between the behavioral improvements and the levels of both activation, in different brain regions among individual rats. We found the significant correlations between the increase in average speed, the number of entries into the center, and also the distance traveled in the center with the beta values of beta signals, which is a reflection of the activation levels in the septum, DBN, LPA, and some other areas. Notably, all these regions were connected to the medial subregion of the LHB rather than the lateral part. Based on the T2 weighted anatomical images, we delineated the electrode tip location of each rat, which is summarized here. The analysis revealed that rats with DBS sites in the medial LHB and close to the medial habenula, shown as red dots here, exhibited greater improvements in average speed compared to rats with more lateral stimulation sites, particularly those in the lateral LHB and far from the MHB. Here shown as the blue dots here. These results suggest that if we want to get a better antidepressant effect, we should stimulate more lateral part of LHB instead of the lateral part. These results, along with the correlation findings mentioned earlier, suggest that the immediate antidepressant effect of DBS primarily originates from the stimulation of the MLHB and also the antidromic and also dromic activation of the circuits connected to the MLHB. We believe that the findings from this research can provide important insights for the development of future DBS therapies. Furthermore, our research highlights that DBS fMRI studies with graphene electrodes can serve as a powerful platform for translational research, investigating the therapeutic mechanism and the modulatory effects of DBS. So in conclusion, our research has focused on two main directions in the field of brain-wide neuroactivity recording. Firstly, we have utilized advanced microfabrication techniques to develop large-scale electrode arrays, enabling us to achieve high spatial temporal resolution in our recordings. Secondly, we have explored the combination of other modalities such as electrophysiology with MRI to expand the potential applications of MRI in DBS studies. It is important to highlight the critical roles played by microfabrication and nanomaterials in realizing these advancements. These technologies have been instrumental in pushing the boundaries of our research. With that, I would like to end my talk, and I want to thank my dedicated students and collaborators for their invaluable contributions to this work. And I also would like to extend my appreciation on the funding agencies that have supported the research. Thank you again for the opportunity and the invitation. It has been a privilege to share our work with you, and I'm grateful for your interest and support.
Thank you so much for this amazing talk. So we have a minute or two for a couple brief questions, if there are any. We're running late with the schedule in general, but so if there are no questions, I will save mine for the panel discussion so that we have more time to discuss between all of them. All right?
Okay. All right. Thank you.
Well, thank you so much again. And yeah. Okay, there is a question in the Q&A, but we will save it for the panel discussion because it applies to some other speakers again. So with this, it is my pleasure to introduce to all of you to the last speaker of this first session. His name is Michael Krieg, and he is a group leader at ICFO, where he is a professor, if I'm not mistaken. And so he's presenting today a talk with a very captivating title. You can see it already, but the title is Neural Engineering with Photons as Synaptic Transmitters. So, Michael, the room is yours, and tell us about photons as synaptic transmitters, please. Thank you so much.
Thank you very much. So I trust you can see my talk and hear me well. So at first, I want to thank the organizers as well for inviting us and giving us the opportunity to present our most recent effort to engineer synthetic neural networks with photons as synaptic transmitters. So my name is Michael Krieg, and I'm a fairly young group leader from ICFO, the Photonic Science Institute in Barcelona, which is on the northeastern part of Spain. This is our institute. And it's not so close, not so far away from Dipsy in the Basque Country, actually. And my group is interested in works in the domain of mechanobiology and optogenetics. And in the mechanobiology part, we are very interested in basic science and the fundamental principles of how cells and animals measure mechanical forces during mechanotransduction, sensory mechanotransduction, for example, the sense of touch and proprioception. So we've heard already in the first talk about the transducing molecules, the ion channels, and we're specifically interested in how these ion channels actually receive the stimulating input. To answer these questions, we developed a mechanical toolbox that spans several orders of magnitude to measure and apply mechanical forces on the level of single molecules in living animals using genetically encoded tension sensors or optical tweezers, but also MEMS and microfluidic devices to apply forces to entire animals and tissues. So at the same time, we can then also read out the deformation applied by these forces and over, again, several orders of magnitude. Our overarching goal with these methods is, of course, to understand not only fundamental principles, but also pathological situations that arise due to, for example, a perturbation of the mechanical properties of neurons and the way how they sense external forces. So in contrast to our other speakers, we apply most of our techniques and our questions to genetic model organisms such as C. elegans, the worm, Drosophila, and zebrafish. But the purpose of today's presentation is slightly different, as we are interested in the engineering of neural networks using novel techniques on neurotechnological diseases. And I don't need to introduce to this audience that the synapses in the brain and neural networks are really the core units where computation happens, and is central for our, let's say, intellectual complexity. So the human brain has a sheer number, uncountable number of them, and that perform all sorts of tasks. And it's quite obvious and understood that a defective neurotransmission leads to a variety of neurological disorders. But it has been recognized early on that actually external interventions through a variety of means, for example, magnetic neurostimulation, electrical stimulation, pharmacological interventions could alleviate potentially some of these diseases and mitigate the burden on the patient. And more recently, also optical interventions by the use of light has been added to this toolbox, and this is going to be the topic of our talk. Why light? In several advantages are conceivable. For example, I mean, it penetrates biological tissues at fairly non-invasively, unless it's encased by a skull, and it comes at various frequencies and wavelength that allows us to use different receptors, which is one of the core advantages as biological systems evolved specific receptors to respond to light. For example, the ones in our eyes or the ones that we're talking about in this talk. So the idea to use light for stimulating neural activity is quite old. It's been proposed in the late 70s by Francis Crick to use light to activate neurons. But still, it took a few decades until the first application or demonstration was available, which used a heterologous system, orthogonal system in hippocampal cells to activate neurons using 3 or more different enzymatic actuators, which was still quite complicated to implement, to make it really a wide applicable method. But then it happened really fast, as the first demonstration that a algal ion channel could actually be transplanted into heterologous cells and elicit currents upon stimulation of the light. And some of these ion channels are expressed in algae that mediate phototaxic and photophobic behaviors, not necessarily the algae that you see in this video here, but similar ones. And then it happened really fast that these ion channels were also expressed in heterologous neurons and animals to show that these animals change their behavior upon light stimulation. And how that works is that this ion channel can be expressed in any cell type in the neuron, and it can then, based on the absorption of a photon, open its conductivity for monovalent and divalent cations, or if they're anionic, also to anions, to depolarize or hyperpolarize the membrane potential. So this is a really promising strategy in neuroscience, and but there's one obvious challenge that has been already talked about is, and our brain is normally quite dark, so there's no light in the brain, and the central question by actually implementing or adapting this technology into a skull, an animal with an opaque skin, is how do we get the light inside delivered to the region of interest. Conventionally, this is achieved using light fibers, or so-called optrodes. And here in this picture is an iconograph, an iconic picture with the mouse implanted with an optrode that delivers the light into the region of interest that express the light-gated ion channel. An experiment like these enable studies like the one in this video here, where the mouse is transgenic for a channelrhodopsin in the amygdala, a center for prey hunting and exciting aggression, and the mouse is seemingly bugged by this toy. But then, when the researcher switches on the light, it is delivered to the area, the mouse starts attacking and changes its behavior quite dramatically. So these experiments are very insightful and very informative to decipher neuronal circuits, but they all rely on the delivery of the light through an optrode or similar devices. But nature actually implemented, or basically delivers already, ways to potentially replace the optrode using enzymes that produce light based on enzymatic reactions. And one of these implementations you see in this little video here, which are not bioluminescent dolphins, but dolphins swimming through dinoflagellates, which itself are fluorescent and emit photons based through as a calcium sensitive enzyme that is activated by mechanical shear forces as the dolphins flow through water. So our approach to that field is to take advantage of these enzymes and encode them in the presynaptic compartment of a neuronal circuit that then would emit photons that are sensed and activated in postsynaptic compartments through classical light-gated ion channels. We implement such a system, or we work towards such a system using the small genetic model organism C. elegans that you can see crawling through that window here. It's transparent, which is very good for our purposes to develop and prove such a concept. So we can also read out neuron activity based on calcium indicators. And here's a video where the whole brain has been labeled with a GCAM, and each of these fluctuations signal a neuron activity event, in that case due to oxygen levels and so on. Moreover, C. elegans has a complete as a known wiring diagram with synaptic connections that are very well understood, and circuits that are also functionally beginning to be characterized. For our purposes, we take advantage of a mechanosensitive avoidance circuit that sits in the nose of the animal. So it's basically one of the neurons that innervates the nose tip, and it senses mechanical touch through connections with two interneurons called AVA and AIB that then elicit a reversal behavior. So the numbers here represent representative numbers for the amount of synaptic connections between these individual neurons. So just to provide the level of details that people think about this neuronal circuit. This is the behavior here, what is mediated by these circuits. So as soon as the animal navigates into an obstacle, and it reverses its crawling direction to eventually avoid this this obstacle. The circuit depends on glutamate signaling. So the predominant neurotransmitter is glutamate that then is secreted from ASH and taken up from the interneurons, and deletions of the glutamate in the animal render the system, the organism, completely insensitive to this nose touch, as we can quantify using our pretty crude assay here. We also established a microfluidic device to interrogate the flow of information through the circuit based on a mechanical touch to the nose. And this device is schematically seen here, in which we have a trapping channel where the animal sits, and then a pneumatic actuator that then delivers a mechanical insult to the nose, while we can read out mechanical behavior, neural activity. Right, so this is obviously the nose, the diaphragm is driven into the nose, and then we can read out neural activity using calcium imaging. In these following videos down here, I show you two examples of a wild-type animal that expresses our gecko in ABA, one of the interneurons, and another animal on the right-hand side that is mutant for glutamate signaling. So what you can see is that the left animal, which is wild-type for glutamate, responds to the nose touch, whereas the right one does not respond to the nose touch delivered in this device. We can now repeat this experiment for many different animals and in many different conditions. And in the wild-type situation, we observe a robust neural activity in the sensory neuron, but also in the two interneurons, ABA and AIB. But as soon as we take out glutamate from the animal through specific mutations, then we see that only the sensory neuron, ASH, responds to the nose touch delivered to the animal, but not the two interneurons. So this is our baseline phenotype now, from which we started to finally replace or recover that chemical, this defective chemical transmission with photons through the bioluminescent reactions. So our aim is to engineer prosthetic synaptic interface with photons instead of chemicals. As I said, we use luciferase to emit the photons, and we use not the one from dinoflagellates, but we use one from shrimps, which is the most powerful luciferase known to date, at least to our knowledge. And we can select and make them more efficient by fusing with the fluorescent protein. It's been described and suggested in Suzuki et al. In 2016. In addition, we make this luciferase sensitive to calcium, which means they emit efficiently more photons in high calcium concentrations that are reminiscent for neural activity. For example, in the situation where ASH is active, we get a higher activity of photon emission. So we wanted to see this live in the worm, but our commercial standard epifluorescent microscopes were unable to deliver images at exposure times that are compatible with the swift biological dynamics that happen during neuronal signaling. So we improved a current luminescent microscope and combined this with deep learning algorithms to eventually record activity in moving animals down to 150 milliseconds. And then we observed, for example, here, an animal that is freely moving with a calcium indicator, bioluminescent calcium indicator in the muscles. And what we observe is indeed a higher photon emission at the contracted side of the muscles, where a high calcium concentration occurs due to its activity. So that approach also works in neurons. And so here you can see a few examples. And my student then went on and combined that methodology with light field imaging in order to obtain a whole 3D representation of the scene with a single exposure and bioluminescence, which provides us actually a 3D render, a 3D representation with 200 to 500 millisecond exposure times. And here indeed, we again see the same, where we see higher photon emission at the contracted side, where the muscles signals to the calcium, and the calcium activity follows precisely the curvature of the animal, therefore, the muscle activity. So with this, we are quite satisfied to have seen that these luciferases work well in C. elegans. And then we turn to the postsynaptic side and engineered a channel rhodopsin with a high operational light sensitivity into ABA and AIB. We employed a selective genetic strategy that I don't want to go into detail for this talk, but we achieve the single cell specificity in AIB and ABA, which you can see here, for example, the localization of a tag channel rhodopsin at the cell periphery. Functionally, we can then ask, do these animals respond to light? And here's one video in AIB. And yes, when the light switches on, the animal reverses its behavior, reminiscent or like similar to the endogenous behavior that the animal would do in the nose touch assay. So here's the other neuron ABA. So the light is delivered and they will reverse their direction. We then titrated different light levels into these transgenics and observed the light response of these animals. And if you then fit the data, fit the model with the data, we can then abstract to low light densities and then estimate that these animals would still have a sensitivity to a very low photon count in the stimulation. Photons with synaptic transmitters. So again, so here after the touch to the nose, our neuron comes on. Then we tested behavior, and here in this condition, we have a very nice tool in hand where we can, so to say, selectively add our cofactors and then prove that only the successful reconstitution can then elicit a behavior effect. So our baseline condition looks like this. They respond to 2.5 out of 10 touches. This is the single cell mutation. Only when the animal is exposed to a functional system using the two cofactors, L-transretinal, the photon absorber, and he carries in the photon emitter, then we obtain a behavior response that is similar to wild-type control animals, proving that our systems can indeed elicit a functional connection between two neurons in the circuit. Finally, one of the other questions we wanted to see is, can we actually now connect to neurons that are normally not having a synaptic connection? So rather than just reconstituting a defect, can we generate novel functions? So here we took advantage of a different sensory paradigm by which the animal actually senses food sources, diacetyl, and migrates or locomotes towards them in attractive response that is mediated through chemosensor olfactory neuron AWA. This AWA mediates attraction in an experiment that looks something like this. So here's a swarm of animals that get attracted to a food source, and after a certain amount of time, most of the animals are found close to the food source. So we adapted this assay and made it a little bit simpler, where we put diacetyl into opposing quadrants of an experimental arena and control conditions. So we know stimulant on the opposite sides, and then we let the animals move towards their target goals. Not surprisingly, in wild type, all the animals, or most of the animals, are found close to the food source, seen here in the density. And in a situation where the animals cannot distinguish the food source through mutations of the factor receptor ODR7, the animals are equally distributed along the 4 quadrants. Now we couple our chemosensor olfactory neuron AWA to the avoidance circuit AVA and AIB. These are the interneurons that I already introduced you before. And then in this, we repeated this assay, and then the control conditions basically this not emit photons but have all the components assembled. Again, the animals are found in the quadrants of the food source, and only little neuron animals are found in the opposing sites. But when we deliver the necessary cofactors such that this system is functional, then a significantly higher fraction of animals get repelled by the food source and avoid that diacetyl chemical, which is proving the fact that we can connect two neurons together that normally do not form synaptic transmission. So with this system, we think the field is open for exploration, and one of the bigger goals that we would like to study at some point is to now combine different colors of photoemitters similar to different neurotransmitters that are employed by a neural network, so glutamate glycine and so on, using, for example, luciferases that emit in various wavelengths together with generodopsins that respond to these wavelengths. So we explored the possibility to use what you saw as the regular generodopsins that are activated by 470 nanometer light, but also green activated anion generodopsins or redshifted carmine generodopsins. So with that, I would like to end the talk and summarize what we added to the ever-growing toolbox of synaptic engineering, using photons as synaptic transmitters, which has also been pioneered by colleagues in the Center of Michigan University in marine model organisms. And what I told you today is that we reconstituted synaptic transmission in the mutant animals and rewired an attractive to an avoidance response. And what I couldn't tell you due to time constraints is that we also generate a gain of function in a male nose-defensive response in the dimorphic, sexually dimorphic system. And we desensitize an endogenous transmission using anion generodopsins in that same circuit. If you want to read more about this, and the growing use of functional bioluminescence in cell biology, neuroscience, I recommend these literature here. And lastly, I would like to thank the people who did most of the work, which is Montserrat Porta and Neos and Shadi and other people who are not in the lab anymore. So with that, I thank you for the invitation again, and I'm happy to answer questions from the audience.
Excellent. Thanks a lot, Michael. And once again, to all the attendees, panelists, and especially to you, we are very, very sorry for what happened. We don't know exactly what happened actually, but nevertheless, super sorry. So what I would do now is jump directly to the panel discussion, because we only have 20 minutes left before the break that is planned. And so maybe we can, I don't know, like share the questions that people may have, including ourselves. So there was one question to break the ice that somebody did online. And let me pick it up.
Could I ask all the panelists to turn on their cameras, and that way we can
yeah, that's
we
a
can
good
all
point.
see each other. Yes. Thank you.
Thanks a lot, Rafa. So Jafet Kinesi was asking what impact does NanoNeuro or the advances that you have been showing may have in invasive neurotechnology devices, or how do you envision basically your most recent advances in the view of the big picture, let's say, of neurotechnologies, etc. So
yeah, I mean, maybe I can start, or is this so I mean, right, I mean, so I think one of the big limitations in our approach is it requires the knowledge of genetic drivers that then ultimately afford how precise you can install or express generodopsins and the luciferases in specific circuits, especially in pre and postsynaptic compartments. Right. I mean, C. elegans, I mean, that's super well established. I mean, we have very detailed single cell sequencing data from neuron-specific gene expression. So we can really pick a selection of promoters or do intersectional strategies to have a very precise expression pattern. And so in future, I mean, we have to see how easy it would be to then apply a similar specificity to mammalian models. And then obviously, right, I mean, ultimately, I mean, if you think about therapeutic interventions, right, we would like to take advantage of this in clinical settings as well. So well, have to see.
So I have a question actually for Yin Wu. So the way that the mTOR is attached, the orientation of the attachment is not any particular angle. No, it's sort of floating. Is that your idea? So would it be better? Would it be better if you put it at the angle to click open the piezo channel, or is that just too complicated?
Yeah, right. I mean, that's quite interesting question. And then piezo has a lateral tension. I mean, there are 3 blades, and then you have a pulling. Lateral pulling is supposed to be opening the gate. So any force, more like horizontal force. So I think that the particle is tagged into, I think, edge of the blade. That's the position. And then the particle, the position is just floating on top of it. And then this particle has some geometrical orientation toward to the magnetic field, and then depending on the location of the particle on the cell with respect to the magnetic field line, I think that matters. So we have done kind of a simple back of the envelope calculation. Basically, it is cosine theta. The angle is the force is a vector. And then you have to have a minimized force, and then as long as it goes over 1.5 piconewton, then it will excite. So the force that we are delivering in ideal condition is about 2.5 piconewton, based on our simulation and also measurement. The force indeed delivered into the system is in average, about 1.5 piconewton. So it matters, and then we are interested in whether where you want to put the magnetic particle. I think matter, and there is also central position. There is something called the cap, and then you can have a tag on that, and then you just indeed rotate. And then people suggest that it will also open the piezo. So that part is still the orientation of a nanoparticle, the field line, and then the exact force exertion is not clear yet. But we have some proposal, and then we should work with biophysicists with a more careful experimental design and so on. So yeah. But nonetheless, we clearly see very reliable, robust calcium ion influx upon the magnetic stimulation with our mTOR.
Thank you. Yeah. I had a quick question for Xiaoji. Do you record each carbon nanotube independently? So do you have can you measure each nanotube independently, or do you have some sort of global mesh? I think you're muted. You're muted. Yeah.
It's actually a film, not individual tubes.
I see.
Yeah, it's a network. Entangled from a network.
And what is the spatial resolution of that field?
Oh, so it's we use the film, but we use the dry etching process to pattern it. So we can make the recording sites. Many normally we use it like 100 by 100 micrometers, but we can decrease it to like 20 by 20 micrometer square. Yeah.
Yeah, and here's a question from someone who knows no physics. So why do you have an artifact in the fMRI from graphene? Wouldn't graphene be all carbon? That shouldn't give you any artifact, no.
Right. And actually, they are not magnetic, but there is a mismatch in the magnetic susceptibility. So because tissue has a magnetic susceptibility which close to water, but if you have implants which has a very different magnetic susceptibility, it will produce artifact. So that's why we choose graphene, because the graphite has a magnetic susceptibility very close to water, but the platinum is very different from water, so it produces a very huge artifact.
Yeah. Thank you.
You're welcome.
Yeah. Excellent. I have a question for John. John Wu. Am I saying it right?
Yeah, Jin, yeah.
You were showing how you could attach the magnetic nanoparticles using bioorthogonal chemistry to some specific channels. How specific can you get? Can you target different channels? Can you target different neurons? What can you do currently?
Yeah, I mean, and there are two different ways. One is using Krylov speed system, and people always utilize, and the other one is using an antibody and kind of antigen or protein tag interaction. I think those are the tricks that we are exploring, and then those are working in combination very well. So that's what we showed. So the particle, nanoparticle can mimic the molecular system by coding some targeting moieties, and that's well known chemistry based approach. And the other one is using the biotechnology process, the cell specific, and those cell type specific, the biotechnology. So I think that's what you utilize.
Yeah, excellent. And then there is, let me check. There are a couple of questions from the audience. One coming from Mohabit Vidiantomo. And it's the $1 million question. He's asking about the blood-brain barrier and how do we plan to overcome it. I mean, he was asking it specifically for Xiangyi, but we can extend it to everyone basically. So how do you foresee this challenge of overcoming the BBB?
For the implanted electrodes, we don't need to overcome the issue. BBB, we want to preserve it. Actually, we don't want to destroy it, especially for the shape-changing electrode array. When we first did the trial, we were worried about if the deployment process will irritate or destroy the blood vessels. Actually, but we did some series of characterization using MRI. We find actually it's quite safe when the deployment happens, because the electrode and also the actuator is somewhat soft, and so basically it cannot destroy the blood vessels. And also we use the several strategies. For example, we injected the animal with the mannitol, which is a reagent can decrease the cranial pressure. So by doing that, we can create a larger space between the skull and the brain, so that the deployment can happen smoothly. Yeah, that's my answer for for this part.
So I have a question for Xiaoji. I really enjoyed your talk. Beautiful. One question is about clever approach on the electrode simulation, graphene-based simulation, electrode simulation, and at the same time measuring the functional MRI, and I see clear evidence that it is working very well. But I mean, some of the challenges that people have encountered is the the spatial resolution of fMRI, and do you really see neural circuit decoding, or do you see new understanding of neural activities upon the simulation of graphene electrode? And do you see something different, or do you see something consistent with other methodologies? Like there are optogenetics plus fMRI studies already there, and how do you compare your with others?
Yeah, what we did is so normally it's a lot more like event-related response. So we inject pulses into the target of the brain, then we use fMRI to map the activation, which area is active. I think the resolution is regular fMRI resolution. It's like millimeters or sub-millimeters. So the resolution is not high compared to the regular technique. What we did is more like, so we combined the behavior test and the fMRI studies. We use I think different from other people in the literature, we use the same set of animals to do behavior tests and fMRI studies. This way we can correlate the fMRI responses with the behavioral changes. I think the main innovation is when we did it together, we can know, for example, for this animal, the therapeutic effect is better. We see how it correlated with the activation of different brain regions.
Right.
This we can get a better idea how this stimulation works, or what is the mechanism. And so compared to optogenetics, I don't think stimulation is more like electrical stimulation. It doesn't have specificity, cellular specificity, but I think it's can better related with the research in humans, because in humans we use electrical stimulation to treat diseases. Right. Yeah, that's my idea of this.
Okay, so you found that the graphite electrode is the optimal or best electrode system that you could pursue?
Oh, well, we are also working on some other design of electrodes. The best thing about graphene is has very small artifact. We can image every part of the brain. We don't worry about the artifact, but it has relatively, at least for now, it has relatively low channel number. And we really want to do a high channel number stimulation with, like, my big dream is to do this like spatial temporal programming of the brain stimulation, but that will need very high number, high resolution stimulation. That is what we want to work on, but we have not there yet.
Okay, great. Wonderful. Thank you.
Thank you.
I have a question for Michael. So it's very impressive, by the way, what you've done. Very courageous. But what would be the ultimate goal? You just want to borrow the wires and replace all the synapses with something that you like better?
Yeah, exactly. No. I mean, so we set out to take it on as an intellectual challenge. I mean, so it was not really. I mean, we do it in C. elegans, and we can't really pretend we're going to cure any disease with this. But obviously, I mean, I would be inspired by people adopting this technology and making these tools faster and better and more efficient, which might actually be a good complementation to traditional optogenetics that require external light, right? I mean, so that is somehow a dream goal. I can't really say that we are actively pursuing this because of all the impeding challenges that lie ahead, but it is a possibility that is imaginable.
Thank you. Thanks.
Yeah. Related to this thing that you were saying of making your techniques faster and better. So I know many people in ICFO, I have many friends there, and I know that there are many people doing nanoparticles and nanophotonics in general, like plasmonic nanoparticles, etc. I'm thinking of Nick Van Hulst, Maria Parajo, etc.
Yes.
Have you thought of applying these sort of optical antennas to try to make your methods more efficient? Because these things are very close, 10 meters away. So
Right. Yeah, no, it's a good question. I mean, the honest answer is I have not, because also I don't know how to get these antennas close enough so there would be an effective way, right? I mean, so, but perhaps, yeah, I mean, it's a different way to think about it. So maybe we should just get together and think that through. I know that people use upconverting nanoparticles for similar stimulations. Yeah, so they basically inject these nanoparticles into the mouse brain and then stimulate or illuminate the brain with a near-infrared laser in order to get emission from these particles that matches with the channel rhodopsins. And people get really promising results as well, right? Which could be an intermediate strategy to actually put this genetic the enzymes into a translational model.
Yeah, that's great. So we'll keep talking about it, because maybe we can help you do that if you're interested. So
sounds good. Yeah.
I had a question for Shigeki. Maybe I missed it, but have you done the chemical labeling on tissue in vivo?
Mm-hmm.
Not on cultures, but in actually tissue in vivo?
Yeah. Now we succeed in chemical labeling of the amplifier in vivo. In this case, our reagent does not penetrate the blood-brain barrier. So we added the reagent by direct injection in the brain. So we succeeded in the chemical labeling of engineered amplifier in the live animals.
Thank you. And have you done it with the NMDA receptor?
We have also succeeded in the visualization of NMDA receptor. However, the specificity is not so good compared with the AMPA receptors.
Thank you.
Thank you.
excellent. So according to the schedule, we should wrap up this first session of the conference. And I think it's a good moment, unless somebody has some very urgent question that cannot live without an answer or something like that. So let me use this minute to thank to all of you for your amazing talks. Once again, apologies for the technical problems that we had. And for all the attendees and for you panelists as well, we're connecting back in 30 minutes. We have a break now of 30 minutes, and we will start with the second session then. So once again, thank you very much, and I hope to see you soon in a minute. Okay,
thank you.
See you.
Let's get started. Here we go. So welcome back, everyone. We had exquisite science in the first session earlier today with 4 phenomenal speakers, and happy to tell you that we have another 4 excellent speakers that hopefully will blow us away with killer science as well. So our first speaker is Juliet Gopinath. She's a professor of electrical, computer, and energy engineering at the University of Colorado Boulder. I think it's fair to say she's a polymath. She works on photonics and quantum engineering and super resolution and 2-photon microscopy. So all of the above. The title of her talk is Shedding Light on the Brain, Super Resolution, and Multi-Photon Microscopy. Juliet, you have 25 minutes.
Yes, thank you. Thank you so much for the kind introduction, and also for the invitation to speak today. It's really a wonderful opportunity. So hopefully you can see my slides. Are the slides okay? Can everybody see? Okay, so I'm with the University of Colorado Boulder, so it's actually still fairly early in the morning here, but I understand it's all different times all over the world. So I wanted to just start by saying a few words about my group. Today, I'm going to be talking about the nonlinear and super resolution microscopy down here, and also our electrowetting adaptive optics. But we do work on ultrafast lasers and integrated optics, as well as time-resolved spectroscopy and quantum technology, as was alluded. So what I'd like to tell you about today is I'd like to tell you about two topics. First, this orbital angular momentum and super resolution microscopy. And secondly, adaptive devices for multi-photon microscopy. And, you know, the focus of my work has really been on developing technology that can be used to image live, awake, behaving animals over a period of time, rather than looking at anesthetized animals and then repeating. So if you think about live animal imaging, and I don't have to spend too much time on this given the audience, but it's really quintessential in being able to image, neuronal activity in alive behaving animals. You see some examples over here. These are miniature microscopes developed by a variety of groups. They're all, I believe, multi-photon or single photon microscopes to allow us to get at some of this information. Most of them function based on looking at fluorescent markers that are sensitive to calcium. And as I alluded to, it can be achieved with either single photon or multi-photon modalities. Now, clearly, this is a tall order. You have to think about the footprint, the weight, the field of view, and the resolution. And so my point here is that there's actually still quite a bit of space for development of new tools for structural imaging. Let's see, that did not do what I wanted. See if I can get rid of this. So let's see if I can.
Do you wanted to advance the slide?
I'm trying.
Oh, there we go.
So,
yeah.
So if you think about stimulated emission, depletion microscopy, and neurology, what you're looking at the bottom of this slide is a neuron, but you can see that the structures that are coming off the neuron are not resolved. So the focus of the first part of my talk will be on super resolution microscopy that allows you to resolve structures like these on a neuron. And these happen to be dendritic spines. So, these are really key for a number of neurological disorders and also neurological understanding. Probably one of the biggest challenges facing our generations is the prevalence of Alzheimer's. And this could potentially provide some insight. And, the point here is that many structures, not just these dendritic spines, really can only be fully resolved with super resolution imaging. So I want to spend a few more minutes talking about the motivation for our FiberSTED. This is still a work in progress, but the idea here is that we really want to study a functioning living animal. And so essentially, if you look at current state of the art, it's below, where you use a big microscope and you anesthetize the animal. What we would like to do is we'd like to combine this with the fiberscopes that I showed you on the previous slide. This happens to be a multi-photon one, so that we can actually really study over a period of time what happens, either with single photon or multi-photon microscopy. So I don't have to spend a lot of time on this, but essentially, if you think about fluorescence microscopy, you scan the sample, you excite the fluorophores, and you're stuck with a resolution of the wavelength over 2 times the numerical aperture. And so the question is, what can you do about this? And so the key is to look at stimulated emission. So rather than the spontaneous emission, that I was talking about, you can use stimulated emission to actually de-excite the fluorophores. And so this is an example. You come in with a photon, and then you get two identical photons out and you de-excite your fluorophore. So how does this play into super resolution? Essentially, here is the main concept. So you have an excitation beam that's Gaussian, and then you have a stead pattern that's of a slightly different wavelength. And what's really important is that the central null be 0. And then when you combine them, you get an effective point spread function that's much smaller than what you started with. And so now your resolution, instead of being stuck at this lambda over 2Na, also scales as the ratio of the stead power to the saturation power of the fluorophores. So it's a pretty powerful technique that can increase your resolution by an order of magnitude. You are limited by how much power you can put on the biological samples, but I think you can still do quite a bit. So just to recap, if you think about confocal microscopy, when you excite a lot of fluorophores, and so you get this fluorescence that's a little blurry. But in the case of STED, the big difference here is that we use this depletion beam to deplete the fluorophores, and so we're able to get much higher resolution. So I'm now going to talk about exactly how do we implement this in a miniature microscope with a fiber tether. And so people have been very interested in using light with orbital angular momentum. And the reason is because it's got this donut shape, and then the phase wraps around as 2 pi, and there's a phase singularity in the center. So this means here that this beam has a very, very dark null. You can think about the wavefronts as being helical. You get plus or minus h bar per photon, and you can describe these beams mathematically as Laguerre Gaussian beams. So the question is how to do this. So, but before doing this, this is actually looking at interfering with a spherical wavefront so that you can actually see the helical wavefront here. So if you think about OAM and fiber, there are a couple of options. So one option is actually to make custom fiber, and that's what's done here in this nice work by these two groups, where essentially you have a ring core. So this is looking at the refractive index versus radius, and this supports the OAM beautifully. But the big problem is that, this isn't available commercially. And I like to joke with Siddharth that, okay, this fiber is going to be like $100,000 per meter. And so we wanted to do something that was maybe a little bit more sustainable. And so one way that you can do this is you can essentially add up modes in fiber. And what you're looking at here is you're looking at the refractive index, and you're looking at the allowed modes. And what you see here is that you essentially, for conventional step index fiber, the first higher order modes are 4-fold degenerate. And it turns out that you actually only need to add up 2 of these modes. So this becomes really problematic. Essentially, you're going to end up with a lot of trouble with sensitivity to bending. And so we actually have chosen to focus on polarization maintaining fiber because of these issues with the step index fiber, that you have these many near degenerate modes and you get this crosstalk. So this is looking at what happens, as to how these things can add up to give you these LP modes that move around. And this really is not a viable way to do things. But if we think about the polarization maintaining fiber, this is fine because the stress rods actually lift the degeneracy. And so now we only have these, 2 well-defined sets of modes for the first higher order mode, they're LP-like, and then also for the second higher order mode. And so we're going to focus on these, but essentially if you add these up correctly, then you can generate orbital angular momentum. So if we want to do that, we also need to be able to quantitatively measure it. The picture I showed you of the wavefront is quite nice, but it provides you with a pretty picture, and you don't have a quantitative sense of how well did you do. And so what we did is we borrowed a technique from laser beam characterization called the twist measurement. And so here, what happens is you have this cylindrical lens, and you know that it's actually very sensitive to the phase of the light that comes in. And so essentially, depending on whether you have orbital angular momentum or you don't, you focus differently. And then using the covariance, you can actually extract out exactly how much orbital angular momentum you have per photon. Because you can imagine for something like STED, if you're combining these beams, you have to do it right. And you have to do it with high quality. So this was looking at continuous OAM tuning in PM fiber, where we added up 2 of these modes. And you could see here this beautiful oscillation between plus and minus 1 as we add up the phase of these modes. Okay, so that's great. You can use this for sensing, but it's maybe not exactly what we want for the STED. So let's talk about what to do. So, there's been previous STED work in standard fiber and also custom fiber, but the problem is here. So essentially, as this phase changes, this mode shape changes all over the place. And the question is, what are you going to do about it? Because this is really, if you do this, you'll essentially be in line with what's already being done. And so the solution actually we came up with by accident, which is to add up the modes incoherently. So here I'm plotting the degree of coherence versus the relative phase. You can essentially see that my shape of my beam changes very drastically depending on the phase. But as I lower the coherence, essentially I can just generate a donut. And so if I do that, then I can basically make a donut. It's not with OAM, but it doesn't depend on bending. And so this will allow us to be able to do this fiber coupled STED microscopy. So this is our setup for doing that. We have an excitation laser at 485, and then we have our STED laser at 585. This is all 1 photon excitation and depletion. And essentially here, we have the beam conditioning to make the donut. We then inject this into this polarization maintaining fiber, focus on the sample, and then we collect from the sample back through this fiber onto a detector. And if you're wondering what's in this beam conditioning, it's actually quite simple. We have a spatial light modulator and then essentially split into 2 arms so that we can rotate one of these patterns by 90 degrees and then combine them again. So this is looking at essentially scanning 80 nanometer gold beads through the focus, and it shows you that we have really nice focus for both the excitation and the STED, and then also in the Z. And so this was our first indication that what we've done would be good enough for STED. And so here you're looking at imaging 45 nanometer fluorescent beads, and you can clearly see that there's a difference in the full width half maximum. And so we're able to get STED resolution that's quite a bit smaller than that of the confocal. And then we also wanted to triple check our dependence on bending. So this is looking at essentially testing the resolution of our system as we bend this fiber. And so here is a box whisker plot and the minimum bend radius. What I want to draw your attention to is that there is a little bit of drift in the system. So we initially did 50 millimeter bend radius and then came back and did it. So essentially what this plot is showing you is that we don't have any dependence on bending. So these are some results from tubulin and HeLa cells. I want to acknowledge that these cells are from Henrietta Lex. And this was especially significant because it was the first data images of biological samples collected through fiber. So we sent the excitation and the depletion beams through the fiber and then collected them back. And then similarly, we wanted to improve. So we wanted to be able to also image deeper because the brain is 3D. And if you look at single photon excitation, you're limited to around 50 microns in depth. But if you use 2 photon excitation, you can go down to about 90 microns in depth over diffraction limited imaging. So essentially here, what we're proposing is to use 2 photons for excitation. Now, For the depletion, we continue to live with the 1 photon depletion because otherwise the power becomes too large for the sample. And this is showing you some of the advantages of the 2 photon excitation. So this is focusing through a tube of fluorescein, and you can see the single photon excitation produces signal all the way across. But in the 2 photon case, you actually have optical sectioning. So you only produce a signal at the focus. And that now means that you no longer need to use a pinhole in your imaging. And then what you're looking at on the right, this is actually looking at imaging a shark, choroid plexus, with 1 and 2 photon imaging. So essentially our change here, the main change here was basically to replace our excitation laser with a Ti-Sapphire at 915. We left everything else the same. The other advantage of using the 2 photon imaging is that the longer wavelengths do not scatter as much through the tissue, so you can go further. And so if you look at these fluorescent beads, you can see that we get almost a factor of 2 in terms of resolution. And then here's some imaging of actual tissue showing that indeed we are actually seeing improvement in resolution. So I want to touch on a few subtle points before I move on to the second part of my talk, which is that if you think about this polarization dependence, you have to be pretty careful as to what happens. So in terms of our donut looks like this. So basically each of the lobes is linearly polarized, but it's got to be linearly polarized in the direction of the null because we're actually focusing very tightly. And so we can no longer use the paraxial approximation. And the question really is what happens if you use a linearly polarized excitation pulse? And so you essentially overlap with these linearly oriented fluorophores. And then the question was, well, what happens if we actually send in 2 pulses that are orthogonally polarized? We can do this by launching at 45 degrees to the PM fiber. And what we see now is we actually see reduced a reduced signal. But that's what's happening here. So I guess I'd like to now summarize this section on the STED. I've showed you these incoherent donut fiber modes result in bend insensitive operation. We have a number of firsts that we're really excited about, both in terms of 1 and 2 photon STED. So I'm now going to move on because I don't want to not talk about this as this is a NanoNeuro symposium. I want to talk about our adaptive optics for multiphoton microscopy. And I don't think I need to spend too much time on this, but let's see if I can go back to my mouse, maybe. But anyhow, this is a video that is showing you what happens as a function of looks like it's not playing, but essentially you can get a very high quality image of a lung this way using multiphoton microscopy. And additionally, you can get some chemical information. So this is exciting. And then what you're looking at here, you're looking at some of our adaptive optical devices. So this is from A Bug's Life, and it illustrates one of the properties of these liquids. They're ultra smooth. And if we use this technique that is actually from the 1800s, it's called electrowetting, where we put a droplet on top of a hydrophobic surface and a conductor, can essentially apply a voltage and change this contact angle. And then the molecules all rearrange themselves for a perfectly spherical surface. And so this is fundamentally a capacitive effect. So it has low power consumption. It doesn't have moving parts. And it's perfect actually to integrate with the microscope. So here, what you're looking at is you're looking at one of our water oil lenses, and you can see this tunes many, many diopters. Now, because we work with liquids, we're actually forced to do some of the engineering right away, which is we have to be able to package them. And so these are some of our current devices where we have them in a glass tube with functionalized sidewalls with electrode, dielectric, and also hydrophobic layer, and also connect it to circuit board. And additionally here, we can also, if we split the sidewall electrodes, we can also make prisms. And so this is quite exciting. and allows us to make these tunable optical elements. So now what I want to do, and let's see if I can make these play, but I want to show you, I want to see if I can show you this large angle scanning. Yes, you can see it now here. So we are actually able to make devices that scan almost 26 degrees, and this is a 4mm device. Then if we go to this one, let's see if I can get it to play. Essentially here, this actually shows you that you can get almost 180 degrees of scanning at 200 Hertz for a LiDAR system. And then at the bottom here, you see this high contrast total internal reflection switch showing almost 70 dB of contrast, which is important for chip scale atomic clocks. So I guess we'll see this video here. So this is a pretty powerful technique for microscopy. And so now I just want to hit some of the highlights of our group. So we've been looking at making these miniature microscopes and incorporating these electrowetting lenses. So here you see a schematic of our first one, and this was done in collaboration with our collaborators at Anschutz, Professor Emily Gibson and Diego Restrepo. So we have a laser, a beam scanner, and then here's where the miniature microscope starts. We have a fiber bundle, and then after that, we have some lenses and include an electrowetting lens. And so this is looking at doing some imaging at different depths of just fixed cells. But then we are able to extend it so that we could actually do live imaging. So this is looking at a picture of one of our miniature microscopes sitting on top of a mouse, and then actually being able to generate this live animal imaging data using the calcium signals. So we also looked a little bit at incorporating our devices for scanning. So for scanning, I mean, you can scan the fiber tip, you can use a MEMS mirror, as we're well aware, or you can use galvos. You know, all of these things work well, but, you know, everybody would like something more, and they're not always compact, especially the galvos. And so we actually wanted to take our electrowetting lens and actually use it as a scanner to replace one of the galvo mirrors. So that's actually what happened. This is, I guess, another schematic of our experiment. So we kept a fast axis for the galvo scanner and a slow axis for the electrowetting prism. And then here are some of our results. Essentially, this is showing you the galvo scanning, the prism scanning, and the overlaid scans. So you can see that these things actually line up very, very well. And then this is actually looking at a picture of these devices. So I guess the last thing that I wanted to mention, and obviously there wasn't quite enough time, but I also have been looking at ways to make more compact and cheaper lasers for doing neurophotonics. And so this is actually a 976 diode-based laser, and it's showing you that we can image beyond 700 nanometers. And the idea behind this diode laser source is quite simple. We wanted to be able to make something that was compact and also produce the power and pulse width that was needed without causing undue expense. Because, you know, using a Ti sapphire laser, it's big and it's also quite expensive, and it requires quite a bit of maintenance. And I don't have this picture here, but my student had drawn a picture of a mouse with a backpack. So that's kind of our dream, so that we can eliminate the fiber tether. And then this is actually looking at some hippocampal neurons expressing YFP. We took our initial work and then extended it to make higher power femtosecond pulses. So with that, I'd like to conclude. I've shown you that with the STED, we can add up incoherent modes and we can get bend insensitive operation. We've demonstrated fiber 1 and 2 photon STED. And then if we think about the multi-photon microscopy, the big achievement was being able to do the live animal imaging. And our devices operate at hundreds of hertz of speeds. They have 28 degrees of steering with these high performance liquids. And we can use non-mechanical scanning if we want and combine them with compact lasers. So with that, I'd like to thank my group. And then also we have a larger collaboration. So this is the CU neurophotonics team, both at Boulder and Anschutz. And then I've just highlighted some of the key contributors to this work. I guess I'd especially like to acknowledge Brendan Hefferman, and then also Robert Niederreiter for their integral roles on the OAM. And then my collaborators, Professor Diego Restrepo, Emily Gibson, and also Professor Bright at CU Boulder. I'd also like to acknowledge our funding. We have funding internally from CU Boulder, also the National Science Foundation and the National Institute of Health. And so with that, I think I'm basically right at time, and I would love to take any questions that you might have. Thank you so much.
Thank you, Juliet. That was very impressive. How far are you from in vivo fibrostat? Yes, so that is the question, right? Because making this miniature objective is not for the faint of heart. Yes. The stat is very, very sensitive to aberrations, and you got to be very, very careful about this. So we actually have some preliminary designs, and we're currently doing testing. And so we should know more very, very soon. We've also been looking at the impact of tissue on STED as you propagate through the tissue. So we had a conference submission on that earlier this year, and we're working pretty hard to do experiment there. Wonderful. Another question I'm just dying to ask you is this new diode laser, would it be commercially available, and what type of price range do you think it would achieve? So we did try to start a company on this in 2018, but we had to stop. We do have patents on it that are available for licensing. Currently, we haven't pursued this further. I guess I wanted to highlight some of the work of Mingji Yu at University of Southern California. So she recently gave a talk and discussed actually putting most of the components that you need for the time lens laser on chip. So I think I'd like to have a discussion with her as well. But I think things are moving in the right direction as far as that is concerned. I guess for me personally, I felt like my heart was in the science. And so I just couldn't manage trying to do commercialization and also be a scientist. Yeah, that would be a tremendous, could have a tremendous impact for the field, because most of us only need monochromatic lasers. And it's just the price of the Thai sapphire. It's essentially a huge energy barrier for people to get into live imaging in vivo. So yes, yeah. I'll follow up with Minji, because she's done some very, very nice work, and maybe we could have a discussion and circle back. Great. Any questions from the audience or from any of the other panelists? Juliet, you should stay for the panel discussion that we're going to have at the end, and you may get questions also then, if they're not coming through now, but they'll pepper through the rest of the session. Okay, so why don't we move on to our next speaker, Lisa Poulikakos. Lisa, if you could turn on your camera. Wonderful. So Lisa is assistant professor in mechanical and aerospace engineering at UC San Diego. She's well known for her work on nanostructure surfaces, which are known as meta surfaces that enhance the scattering of light, and she's going to be talking about that. Nature-inspired colorimetric meta surfaces for next generation on-chip imaging of tissue microstructure. So Lisa, you have 25 minutes, and then we'll have 5 minutes for questions. Thank you.
Well, hello, everybody. Thank you so much for the kind invitation to present my work here today at this exciting workshop. My name is Lisa Pouliakos, as you said. Today, I'd like to talk to you about how nature-inspired colorimetric metasurfaces, such as those that you see in this image here, hold immense potential as the next generation for on-chip imaging of tissue microstructure. So, photonics, and particularly nanophotonics, holds immense potential to address global health challenges, where here I'm highlighting some exciting recent examples from the field where metasurfaces have been used to interact with live cells. They've been studied to detect extracellular vesicles. They've been investigated for bacterial bloodstream SIRS detection, molecular barcoding. There's some recent really nice work on photothermal tumor ablation with gold nanoshells that's in clinical trials, or even experiments with optical tweezing at a distance. So building and expanding on this exciting body of work, my lab develops new classes of chiral and anisotropic nanophotonic materials for a series of next generation imaging applications. So here you can see 3 branches that my group currently focuses on. The first looks at nanophotonics for molecular disease treatment, where we study the handedness or chirality of small molecules. In a second branch, we're investigating nanophotonics for imaging tissue or soft material microstructure to develop new kinds of label-free and contact-free imaging platforms. And in a third branch, we're developing nanophotonics for miniaturized on-chip super resolution imaging, where we're also studying systems which merge the physics of light and sound for this purpose. So today I'd like to focus on our second branch, studying nanophotonics for tissue and soft material microstructure imaging. So in this application, we're particularly inspired by the important health challenge of organ fibrosis, which accompanies major causes of death worldwide, such as Alzheimer's disease, heart disease, and various cancers. However, unfortunately, we currently lack effective diagnostic tools and treatments for organ fibrosis, and in particular, there's an urgent need for new diagnostic tools which quantify the tissue microstructure. So if we look at diagnostic tools that exist today, we have, for instance, immunofluorescence staining or biochemical staining, which provides a wealth of information but does not quantify the tissue microstructure, or tissue microstructure imaging technologies such as scanning electron microscopy, where you can see here that it beautifully quantifies the tissue microstructural properties, but this method is destructive and costly and therefore not widely available. So our research aims to address these challenges by developing a new class of on-chip metasurfaces, which are able to map tissue microstructure onto color in a precise, low-cost, non-destructive, and label-free manner. So how can we do this? We like to draw inspiration from nature, where the beautiful blues that you see in the morpho butterfly wing arise not from the absorption of light and color pigments, like most of the colors you see around you, but instead these blues come from the scattering of light off of its micro nanostructured surface in a phenomenon known as structural color. So we're particularly inspired by 3 properties of the butterfly wing. First, its nanoscale features enable strong interaction with visible light. It's chemically selective, meaning that we can move from an air to an ethanol environment and drastically change the color response. And finally, what's most important for our work, the micro nanostructured features of the morpho butterfly wing are anisotropic, so they extend differently across different spatial directions, and this leads to a very clear angle and polarization sensitive color response. So we're working on leveraging 2 key strengths of nanophotonics toward our goal of tissue microstructure imaging, where the first is the ability of nanophotonic systems to generate highly tunable structural color. And the second is the ability of nanophotonics to scale down the complex manipulation of light from a meter cubed, where you typically require a whole laser lab with several bulky and complex components, down to a millimeter squared, where the same complex manipulation of light can be achieved on a single nanostructured surface. So here you can see an outline of our metasurface imaging concept, where we start with our nanopatterned metasurface. It can be patterned, for instance, on a standard microscope cover slip that's used for imaging. Our system is then, our tissue structures will then be illuminated with polarized white light. When the system traverses either healthy or fibrotic tissue, its polarization state will selectively change. Then when it interacts with the metasurface of interest, structural color, varying color responses will reflect back from the system and thereby map the tissue microstructural properties onto a structural color response. So let's dive into the metasurface concept in a little bit more depth. You can see here that we start by designing anisotropic metasurface elements, which can interact selectively with different polarization states of light. Then these polarization-sensitive color responses are rationally designed such that every incident polarization state gives rise to a unique color response. And we can then use this colorimetric polarization sensitivity to map the fibrous properties of biological tissue onto a color, because different degrees of fibrosis in the tissue lead to a variation of the polarization response in our system, and that can then vary our colorimetric response. So how can we design metasurfaces for tissue diagnostics? You can see here that we can build and expand on a whole body of work using plasmonic and dielectric metasurfaces for colorimetry. And the example of plasmonics, here are just some seminal examples of previous work where you can see plasmonic crosses can allow for polarization sensitive color responses, as well as plasmonic systems which have been used as refractive index sensors through colorimetry. In the case of dielectrics, we can consider polarization-sensitive color responses for high refractive index Me resonators, in this case, the cross structures. And in this work, we'll focus on so-called guided mode resonances, which allow for narrow bandwidth structural color responses. So to start this work, we performed numerical simulations of a proof of concept guided mode resonant metasurface system, and we're able to demonstrate numerically the potential of metasurfaces to visualize tissue microstructure in medically relevant cases. So here you see our computational results for the example case of breast cancer, where we can differentiate through the color response of our metasurface system an early stage localized cancer where collagen fibers are tangentially aligned along tumor margins, from a late stage metastasized cancer where the collagen fibers are outward oriented with respect to the tumor margins. So these promising computational results then led us to a series of experimental projects and studies, where today I'd like to talk to you about two of them. And the first is an experimental proof of concept study, which discusses how we can map tissue microstructural properties onto color. And the second is an outlook on some ongoing work of nature-inspired 3D architected metasurfaces. So let's start with our experimental proof of concept. So in this project, we studied a very simple case of guided mode resonant metasurface gratings, which are composed of a glass substrate and a silicon nitride layer, which is patterned with gratings of varying pitches. As we increase the metasurface grating pitch, as you can see here, both in the spectral response and the optical microscope images, we can clearly tune the color response of our system. We then interfaced our colorimetric metasurfaces with polycaprolactone nanofibers. These nanofibers lend themselves particularly well to mimic very controllably the microstructural behavior of fibrous biological tissue. And this is because these fibrous constructs allow us to apply varying strain rates. So by applying an increasing strain rate, we can controllably increase the nanofiber alignment and thereby mimic varying alignments of fibers that also arise very similarly in biological tissue. So here you can see our experimental configuration shown schematically, where because the PCL nanofiber composites are opaque, we have a back reflection configuration where we send in linearly polarized white light through the system. It reflects back from the tissue, and for different fibrous degrees of the tissue, we expect a different polarization state of light to be reflected back, which is then mapped onto a color response in the metasurface. And in particular, we investigate two parameters. First, the system of varying grating pitch, and second, we vary the PCL nanofiber draw ratios and compare their interaction with the metasurface. So here you can see again our metasurface results for increasing grating pitch, both the spectra and the optical microscope images when no PCL nanofibers are present. If we then interface our system with draw ratio 10 PCL nanofibers, you can see clearly here that the color response changes both in the spectra and in the optical microscope images. So how do we now quantify this color response? Here you can see again our optical microscope images with and without the nanofibers side by side. We can see a clear color difference by eye. But we can also quantify this color difference with color theory. The so-called CIE-LAB color theory system allows us to define the so-called color discrimination factor, shown here, delta EAB. This color discrimination factor quantifies how easily the human eye can distinguish two adjacent colors, where 0 is the minimum and 1 is the maximum value. So here you can see our results for the color discrimination factor for our metasurfaces of varying grading pitch, where the color of the histograms is the system interface with the draw ratio 10 PCL fibers, and the color without the PCL fibers is shown on the top, of course. And now we can see clearly, if we compare them, that our 160 nanometer grading pitch shows the greatest performance. So it shows the biggest difference, in a sense, in color response when interfaced with the PCL nanofibers. So we'll now focus a little bit more clearly on our best performing 160 nanometer grading pitch metasurface. You can see here, where on the bottom row, we have our system without PCL nanofibers, the spectra and optical microscope images. Then if we move from the bottom to the top, we've interfaced our system step by step with PCL nanofibers of increasing alignment. So going from draw ratio 1 to draw ratio 10. If we look at these color responses, they look quite similar at first sight, but you can see, due to the sensitivity of our narrow bandwidth guided mode resonances, that there are indeed differences in the spectral response. And indeed, if we compute our color discrimination factor, we can see a monotonically increasing color discrimination factor for increasing draw ratios. So that tells us, as the nanofibers are more aligned, we have a larger color discrimination factors. The color of our metasurface changes more, the more aligned the nanofibers are. So why does this happen? We hypothesize that this has to do with the polarization state of the light that changes with increasing alignment of our nanofibers. So we hypothesize in particular that for linearly polarized illumination, when the light reflects off of the nanofibers, it gains a certain degree of ellipticity. So to characterize this ellipticity, we studied the Stokes parameters. In particular, we studied the third Stokes parameter, which quantifies the degree of ellipticity of the reflected light from our sample. And in particular, you can see here that with incident linearly polarized light, the nanofibers of draw ratio 1 gain a very clear non-zero degree of ellipticity. If we interface the system then with draw ratio 10 nanofibers, which have a higher alignment, you can see that the amplitude of our third Stokes parameter, this ellipticity, further increases. So that means that we have a higher degree of ellipticity the more aligned the nanofibers are. And this assessment of the ellipticity of the light aligns very well with our findings of the metasurface, which showed through our colorimetric analysis that with increasing nanofiber draw ratio, so with increasing nanofiber alignment, we have a larger color difference. And that, in a sense, indicates or maps onto the fact that we have a higher ellipticity in the polarized light reflected from the nanofibers. We can also, through our Stokes parameter analysis, better explain why our 160 nanometer grading pitch metasurface showed the best performance. And indeed, if we consider the resonant wavelength of our guided mode resonance system with the peak wavelength of our highest degree of ellipticity, these are indeed matching. So our metasurface, where the guided mode resonance is mapped onto the wavelength of highest ellipticity, showed the strongest structural color response. So these results then show us very clearly as a nice proof of concept that our metasurfaces are able to detect through colorimetry very distinct changes in the fiber alignment. So they're able to map the PCL nanofiber microstructure onto a quantitative color response. So building on these promising results with our proof of concept metasurface grading system, we can now ask ourselves as the next question, how can we boost the metasurface performance? How can we even further amplify the color discrimination that we use to map the tissue microstructural properties? And for this, I'd like to present an outlook on some ongoing work on nature-inspired 3D architected metasurfaces. So we know that in nature, we have structural color in many cases. Here are some examples of the Papillot Blumet butterfly or the Calcoteus maragdina beetle. You can see here, upon linearly polarized illumination with cross-linear polarizers, we have a very clear structural color response. the structural color response drastically changes upon circularly polarized light illumination, as is shown also in our spectroscopic images. Now, of course, the structural color arises from intricate micro and nanostructured systems we can verify with our scanning electron micrographs shown here, as well as for the beetle shell. So the micro and nanostructured systems differ very strongly in the photonic crystals shown in each case. So this concept we'd like to leverage and build upon by drawing inspiration from this naturally occurring structural color to go from our nature-derived structural color to new classes of 3D architected metasurfaces, which could even surpass the performance of what's seen in nature. And these 3D architected metasurfaces will hold several advantages in enhanced optical anisotropy and colorimetry, looking at unexplored design spaces. So by adding a 3D architected dimension to our metasurfaces, we have whole different classes of materials we can design. We have different design spaces with which we can tailor the anisotropy of our system. And also the 3D architecting allows for larger feature sizes to obtain our structural color effects, which enables potentially more easily scalable 3D architected fabrication. So in order to better understand where the structural color or the anisotropic structural color in nature-derived systems is coming, we assess our systems with two key figures of merit. So here for the example system of the morpho butterfly wing, you can see here our system illuminated between cross-linear polarizers. The butterfly wing is rotating at different angles, and you can see a clearly linearly birefringent behavior as the intensity of the system maximizes and then is completely extinguished later. So to better model this, we introduced a Jones calculus model of the intensity in the system, where you can see here the Jones matrix, where phi b is the phase delay and theta is the optical axis orientation. So then computing the intensity with our Jones calculus model, we get the expression you can see here, and we use that to then derive the phase delay and optical axis orientation and quantify the birefringence of our system. So for the morpho butterfly wing shown here, our Jones model fits beautifully to the data, and we're able to derive the phase delay and optical axis orientation quantitatively, showing that the morpho butterfly wing exhibits a strong linear birefringence. Our second key figure of merit is looking at the polarization sensitive colorimetry of the morpho butterfly wing. So in this case, we illuminated the system with circularly polarized light, and we can see here a drastically different color response where the color transitions from a green-like to a red-like color in this case. Now, in order to quantify the anisotropy of this color response, we can again use our delta EIB, our color discrimination factor shown here. But in this case, we'd like to compare our color discrimination factor of mutually orthogonal orientation. So we'd like to verify that indeed this morpho butterfly wing breaks C4 rotational symmetry. And in doing so, it can be, as an imaging platform, it can really surpass the performance of existing methods such as polarized light microscopy by breaking C4 rotational symmetry. And indeed, by calculating the color discrimination factor for these varying reference angles, you can see here that we have a non-zero value in all cases. So in symmetric systems or C4 symmetric systems like polarized light microscopy, this value would be 0 in all cases. Before the morpho butterfly wing, we have non-zero color discrimination factors where we have at each angle considered, we're looking at the color difference between an angle itself and its 90-degree rotated color response. So this really tells us that the morpho butterfly wing exhibits a strong optical anisotropy, breaking C4 symmetry in its colorimetric response. So inspired by these nature-derived systems, we're now just starting to develop new classes of 3D architected metasurfaces. Here you can see some of our first examples, which consist of just a very simple proof of concept system, which are two stacked gratings placed orthogonally above each other. And you can see indeed for this 3D architected system, we have larger feature sizes than for their 2D counterpart. But with these larger feature sizes, we can obtain a very distinct color response already for this proof of concept system, where for linearly polarized illumination, you can see between cross-linear polarizers very clear increase and extinction of the intensity, showing a birefringent behavior. And for circularly polarized illumination, we see a very clear variety in the structural color response at varying orientation angles, ranging from green to red. And These are just first proof of concept systems that you can see here, which is just the beginning, and we're leveraging this to further optimize the design and even surpass the performance of what is seen in nature. So I'd like to end with an outlook on the health challenge and the importance of the health challenge that motivates this work. So around 1/3 of deaths worldwide is related to organ fibrosis, and this can really affect organs across the human body, from lung fibrosis, which is an important consequence of COVID-19, to, for instance, liver fibrosis, amyloid plaques in the brain as a form of neurodegenerative disease, or skin cancer, and many more examples. And because our metasurfaces leverage light as a non-destructive and label-free probe of the tissue microstructure, they exhibit a translatability across organs and across diseases that is not easily attainable with existing biochemical probes. And I envision a future where these metasurfaces can be integrated in versatile clinical and biomedical imaging applications, ranging from pathology to high-throughput drug screening or in vivo endoscopy applications. And with this, I'd like to thank you for your attention and the very kind invitation to present our work here today. Many thanks to my wonderful students at UCSD and our collaborators. Here you can see us at our latest excursion to the butterfly farms nearby San Diego for some research inspiration, and also thank you to our funding sources. Thank you for your attention.
Thank you, Lisa. This is not only beautiful, but it must be a lot of fun to carry out this work.
Absolutely, it's a lot of fun to work with these butterflies and beetle shells and everything. So we've been enjoying it a lot.
So here's a question for you. So for the fibrosis project, you would expect that different levels of fibrosis will result in different levels of polarization as the light gets into the meta surfaces. That's sort of the logic, and you'll have like an assay of maybe collagen. Is collagen what's polarizing the light in the tissue?
Yeah, so collagen is most often the case. That is most often what is polarizing the tissue, but there are also examples where we could look at amyloid plaques, for instance, so relevant to neuroscience, of course, fitting for this workshop. But also there are systems like actin and myosin, so other fibrous constructs that could also give such an effect.
And where would the light come from? Would you shine light from the outside into the tissue or into the metasurface?
Yeah, so some of the first applications we're looking at are just with histological tissue sections. So basically outside of the body, just for pathology work. But we do envision, as you see on this brief sketch here, future endoscopy applications where we could integrate the metasurface into an endoscope and use that for selective imaging of fibrous constructs in vivo. There's also the opportunity, because it's contact free, to implement it with live cell imaging and tissue cultures and so on.
Wonderful. And then the idea is that you could also have just a human detected by eye, no? Just like
exactly
by color. Yeah.
So you can either use a simple microscope, as is available in any pathology lab or simple medical lab, or eventually it would be great to be able to just detect it by eye as well.
Yeah. Wonderful. So any questions for Lisa from any of the registrants or listening to the meeting, or from the speakers? From Aitzol, you have a question? Or no? Did you raise your hand? No, you're not raising your hand. No? Or maybe you're muted. No, you're not muted.
No, now do you hear me?
Yes, I can hear you now.
Yeah. Okay, I'm sorry. I got a cold while you were talking, and so my microphone got disconnected. Now I'm connected back again. So a super basic physics question or optics question. So I'm surprised about the fact that you get any birefringence. I always had trouble with that word, with circular polarization. So that's the polarization average out somehow, and then you should get anything. I don't know.
So that's a great question. So basically there's an output analyzer. So it's not just the circular polarization itself, because that would be more symmetric. So the output analyzer lets us probe the asymmetry. So
perfect. Now
so you're absolutely right.
Excellent. That's it.
Yes.
Thank you very much.
Yes.
Wonderful. So if we don't have any other questions, maybe we can move on. And Lisa, please stay till the end of the session because we'll need you again during for the final panel, after the keynote. So we started the meeting this morning with an exquisite talk on magnetogenetics. And some of you may think, well, when is that ever going to make it into some sort of human treatment? Well, we're going to have now actually going to from the nano to the macro, a talk on the use of magnetic methods in the clinic. And the speaker is Alvaro Pascual Leone. He's professor of neurology at Harvard Medical School. And he's actually a world leader in the development of transcranial magnetic stimulation. The title of his talk is State of the Art and the Future of Non-Invasive Neuromodulation. Alvaro, you've got the, you have 25 minutes plus 5 minutes of questions.
Thank you. Let me add the thanks of the other panelists to the invitation and the opportunity to present to you. Like Rafa is saying, my goal is to tell you a little bit about the applications of these technologies in the clinic, both what we can do now and also what we're missing, which I think of opportunities to develop and improve on the technology for reasons I'll try to lay out. So just by way of disclosure, these are my disclosures as sort of Harvard likes us to do. I think the most important disclosure is that I will be telling you, in as much as I give you specific examples of applications, but work that is done by talented people I have the opportunity to work with, I'll try to recognize them. But if I don't, assume somebody else did the work. So I'll try to point out who has done what. But I also will tell you about off-label applications because I think it highlights the potential of the technique. So applications that are not really fully approved in the clinic by the FDA, but are being applied in patients or in an experimental setting with IDEs. I'm going to focus on non-invasive approaches of neuromodulation, and there are many ways to bridge the skin and skull and reach the brain. I'm going to talk about one, electromagnetic, as Rafa was saying, but one can apply direct magnetic fields, phonic stimulation, acoustic stimulation, and there are growing number of techniques developing, and it's an exciting, rapidly growing field with differences that allow different types of applications. The two I'll focus on are transcranial magnetic stimulation, which is essentially Faraday's principles applied to neurobiology. Instead of a secondary coil, the brain serves as a sort of an array of secondary coils, and electromagnetic induction bridges the current that passes through a first coil. The magnetic field pulse enters the brain and induces a current. So think about it really as electrode-less electric stimulation via electromagnetic induction. And then I'll tell you about transcranial current stimulation, which again is essentially Faradizing the tissue, creating a gradient of current between two or more electrodes that is not sufficient to depolarize neurons to the point of firing, but it is sufficient to shift membrane potentials and in specific frequencies, even if you apply alternating current and induce specific oscillations and promote synaptic plasticity. At a macro scale, like Rafa is saying, the kind of limitation of this technique is mostly in its spatial resolution, but also to some degree in its temporal resolution. Now, there are a growing number of devices for transcranial magnetic stimulation that are FDA cleared for a number of different indications. Here are some of the pictures of them. In the meantime, these applications, particularly depression, is not only CMS approved, but also covered by Medicare, Medicaid, and a number of insurances in the US, and adopted and similarly covered by healthcare systems in other countries. The real clinical impact is substantial. In ballpark figures predict that about 70% of patients, for example, with medication-resistant depression, will respond with a meaningful, clinically meaningful response. About 30% will go into a full remission. Contrast that against the likelihood of response to any other medication of well below 4%. So it's an order of magnitude more than 25 patients are projected to be going into remission of their disabling medication-resistant depression per day in the United States alone, because of the number of systems that are being developed and applied. In addition, there is a very rapidly growing list of off-label applications where the device that is approved for a given indication is applied for other indications, and there is a growing interest in neurology and psychiatry and rehabilitation for a number of various conditions for which otherwise interventions are limited. One example of a different type of FDA clear application is not therapeutic, but actually diagnostic. One can use non-invasive stimulation guided by frameless stereotaxy and by neuroimaging guidance to map motor output in speech representation, and in doing so achieve a resolution that is quite comparable with what we can achieve through direct cortical stimulation in the operating room setting. And so it's an approved indication that allows to shorten the time in the operating room and make therefore the interventions more efficient, plan them more precisely, but in addition has critically contributed to the development of frameless stereotactic system that now allows even greater precision in the therapeutic applications. So it's a case where the development of technology has led to a particular kind of application, but also improved the others. So it's exciting. We're helping patients. However, I'm going to argue that we're truly only scratching the surface, at the tip of the iceberg kind of thing, among other things, because conceptually, we're really struggling to leverage the real potential of neuromodulation. The promise of neuromodulation, in my mind, is not a treatment for an illness. They are approved for a given illness. I think they're conceptually not that. They're conceptually a tool that allows modulation of specific substrates that map onto symptoms and disability. In a real sense, we're not treating depression, we're improving mood symptoms, we're improving apathy. We are not treating dementia or Alzheimer's, but improving memory function or executive functions. And making that explicit in the targeting and in the conceptualization, I think is worth it. And so I like to illustrate that a little bit and give you a sense of where that can take us. The fundamental notion is that for any emotion, thought, behavior, there is a spatiotemporal signature that we can capture with imaging and high-density EEG, define the correlate of that function, and in the setting of disease, identify the alteration of spatiotemporal signatures that map onto symptoms or disabilities of the patient, almost regardless of the underlying pathology or only prompted by the underlying pathology. But the potential then is that in targeting those altered spatiotemporal signatures, we have the opportunity, as I'll try to show you, to not only improve the symptoms and disabilities, but potentially by normalizing the pattern of functional activity, influence the underlying pathology, which is exciting. So we know that we can apply the stimulus, for example, with transcranial magnetic stimulation, and record the EEG impact using high-density EEG to identify the patterns of responses to different areas. If we can scale down the stimulus and apply a truly translational biomarkers in animal models and gain insight into the mechanisms of action. I won't belabor that point, but I do want to illustrate the fact that what we've come to realize is that the pattern of response of any one individual brain of ours allows, through these perturbations, through these stimuli, to identify a brain fingerprint that is exceedingly reliable in a given individual, despite the fact that it's substantial in the inter-individual variability. And so what we can then do is identify patterns of response to the stimuli that can be followed longitudinally within one subject and detect deviations within that subject that allow to define targets of intervention and targets of treatment, ultimately, if there are symptoms accompanying them. So the idea then is that if we take that potential seriously and pursue it, rather than a non-invasive way with some impact for a treatment of a given condition, a given disease, we might have a real precision medicine approach where we identify symptoms or complaints from a clinical unpacking of the presentation, link that to the identification of the individual physiologic biomarkers, personalize the target, and then monitor the physiologic effects and close loop adapt the stimulation. And I want to illustrate that a little bit to you as the potential future of these technologies. To start with, we can do all kinds of patterns of stimulation, obviously, with transcranial techniques, with TMS or with TCS. There is often talk of these parameters leading to some overall effects, slow frequency patterns leading to an inhibitory effect and an excitatory effect. I think it is a fundamental misunderstanding of the effects to assume that what might be a population net effect applies to physiologic underpinnings or to individual subjects. And so one of the things that the field has been struggling with and has to come to terms with is to stop talking about inhibitory or excitatory LTP-like or LTD-like parameters and realize the fact that the effects, as you see here, might be whatever they are at a given population level, but individually are exceedingly variable. There is no pattern, no parameters of stimulation that are intrinsically inhibitory or excitatory. It is an interaction between the state of activity in the individual brain, the structure of the individual brain and organization, and the parameters of stimulation. At the end of the day, what that means is that we need to measure the effects induced to really be able to fine-tune the parameters. Let me illustrate that with an experiment from Mark Eldaev that is more recent, using EEG and functional imaging, targeting a given location in the angular gyrus and showing that at a population level, high frequency and low frequency patterns of stimulation lead to a very different modification of connectivities and patterns of activity induced after the stimulation. But if you look at the individual subjects, again, you have, similar to the data I was showing earlier from Fumiko Maeda, a very large inter-individual variability of the induced effects, almost completely opposite effects in different subjects, that if you measure and fine-tune the parameters, you can actually achieve a given effect. So in fact, for example, this subject here, if you see my pointer, that shows a facilitation of the connectivity with the low frequency, is the one that would show a very prominent suppression with 20 hertz frequency, the exact opposite of what overall in the population it might be achieved. So the parameters need to be individualized for a given effect if you want to have a consistency of the modulation, and with that, explore at least the consistency of the mapping between modification of physiologic signatures and behavioral effects. So we need to measure we need to monitor while we apply the stimulation, and that enables us to have a closed loop dose control approach. The second point worth emphasizing is that even though we target a given area, and oftentimes people talk about the therapeutic effect dependent on the targeted area, in fact, the targeted area at best should be viewed as a window to a network effect. And that is been shown quite nicely, for example, by Anthony Valero here in animal models. It's harder to do in humans, but with MRI, structural MRI, DTI, we can do similar demonstrations in humans. In this case, you get a targeted stimulation to the visual cortex, and you see, for example, a selective impact at a distance in the splenial visual area, which has to do with the fact that there are rich connections between those two areas that happen to be excitatory. And so if you suppress activity in the targeted region, you end up removing input and therefore reducing activity in the distal area. What Tony was able to show is that, in fact, one can predict precisely that distributed effect on the basis of the strength of the projections and their weighting, as it were. And the same exact effect can be achieved in humans. And in doing so, it provides a sort of roadmap to approach the question of where to target the stimulation in order to achieve a given desired behavioral or clinical effect. I thought I'll illustrate this with a case. I'm a neurologist, so I'm a little biased about the value of individual cases, so bear with me. But this is an interesting kind of presentation. This is a patient who presents acutely with a sudden onset of vivid, well-formed, elaborate, colorful hallucinations. In fact, she was in her bathroom washing her face and saw suddenly a caravan of little elephants walking across the sink, much to her surprise. And her husband took her to the emergency room, where they found that little stroke, an acute stroke in the thalamus. The presentation is consistent with what John Lermit described as a peduncular hallucination, called peduncular hallucination, because of the assumption that there is some sort of release of peduncular activity. But the lesion is in an area that has nothing to do with vision. And Aaron Boas, the resident who was seeing the patient, asked himself, how come these are the symptoms that this patient presents with? And collected a number of cases, 23 in total, that had presented with an acute onset of peduncular hallucination. And you see in red the location of the lesions of those patients. And what you can see is that they are sort of all over the place, clinically exceedingly and exquisitely consistent, but the substrate apparently very different. If you do an overlap of those lesions, traditional sort of morphometric kind of MRI analysis, the best you get is a common overlap between 6 cases out of the 23, with lesions that cluster around the thalamus. But the vast majority of the cases remain, quote, unexplained. It's difficult to explain how. So what Aaron and Mike did then is, instead of focusing on the localization of the lesion, ask the question of what impact the lesion, whatever it is, would have on the connectomics defined functional connectivity of the brain, and develop a technique called lesion network overlap, where the impact of a given lesion of the patient I showed you or others on the network connectivity, it can then be added up and overlapped. And in that case, all of a sudden, you now see a very concrete region showing up in 23 out of the 23 cases, which maps onto area MTB5, a visual area that has to do with well-formed, elaborate, moving objects as we detect them. That allows to localize the symptom, that it is the dysfunction there, which in fact is an augmentation of excitability with a lesion that explains presumably the symptom localization. And we can test this using it as a target for the intervention and targeting that area, the hallucinations in these patients go away. So we can use the fact that the effects are network-based as a means to define the target and guide a personalized targeting. Oftentimes, it's not a single lesion that will come up, but a distributed network of areas, some being more, others less active with a given task. And so instead of using a single focus stimulation, one can use multifocal forms of stimulation, targeting different areas, and in fact, letting the data of the pattern of activity guide that with Monte Carlo simulations to define a pattern of electrodes which map and reverse the abnormal spatial temporal signature of activity. By using EEG guidance, one can use not only spatially precise, but brain oscillatory pattern precise, so frequency tuned patterns of stimulation. With Giulio Ruffini, we defined an algorithm to generate those montages, as it were, those protocols of stimulation called Steamweaver. And when applying them, one can increase the efficacy of the intervention, but it leaves out the question of what about deep structures? What about if you have patterns of activity that include selectively areas not in the cortical surface, but deep in the brain? In order to address that, one of the approaches that Nir Grossman developed with Ed Boyden and myself some years ago is something called temporal interference, the idea of having a carrying frequency at very high frequencies, say 20 hertz, apply the stimulation in a way that more than one beam of stimulation in anti-phase orientation come together at the targeted deep structure and effectively cancel each other out, leaving there a residual difference. So 20 hertz versus, 20,000 hertz versus 20,010 anti-phase, and leaves 10 hertz in the targeted area, and you can orient it appropriately. To be quite honest, when Nir proposed this to me, I thought he was completely out of his mind, that it would never work, that it would be so much dispersion that it would never localize. He went on to do experimental work in mice and showed that it was possible to selectively target and modulate activity in the hippocampus with the temporal interference, which could not be done with non-interfering equal amounts of stimulation. And then the question became, well, can you do that in humans? The heads are much bigger, the dispersion is going to be much greater. It's taken some time, but Inés Violante has completed a very carefully designed study starting from cadaver modeling to very carefully define modeling work with a group in Zurich, and has now a study in press in Nature Neuroscience showing that it is possible in humans to target subfields of the hippocampus and have an effect on memory. So it appears that it is possible to modulate network to one node as a window or to multiple nodes with which can include deep structures using temporal interference approaches. So if you do that, what can you then clinically accomplish? I want to finish showing you sort of the translation. I'm a behavioral neurologist in clinical practice, so I deal with patients with dementia. So I've been interested in the possibility of applying this, particularly to try to improve cognitive function in such patients, in patients with Alzheimer's disease, for example. So the approach we've been taken is define the pattern, identify the pattern and characterize it of the spatial temporal signature associated with their deficits in a given task, use source localization to try to identify the nodes of that network, as it were, that sustains the spatial temporal signature identified, generate an electrode montage to target that network, define it that way, and then send the patient home with the system that allows for home-based stimulation, such that they can do 20 minutes, 30 minutes per day for the course of about a month and follow them clinically. It can be monitored remotely. It allows us to make sure that they're doing the stimulation correctly, and we can assess in real time their performance through web-based applications and tasks. Neuroelectrics is a company that has developed one such system. There are others in the market now. And to illustrate the potential of it, here is results of a group of patients with attention deficit disorder with mild cognitive impairment, performing an episodic memory task. And you see a very clear difference in the pattern of activity electroencephalographically associated with the task as compared to older age-matched healthy controls. The source localization identifies a particular node in the angular gyrus and a connectivity between that angular gyrus node and hippocampal structure. When you target area in a given subject, this is one subject who had shown a very clear, dramatic worsening in episodic memory over the course of 2.5 years. Over the course of 14 weeks, you completely reverse the dysfunction. There's a million questions to be asked. To start with, how long does that last? Can we sustain the benefit? How do we maintain it? How consistent is it? I don't have answers for many of those questions, but at least at a population level, when you look at a study now with 60 individuals per group, getting either the right personally targeted stimulation at the right frequency or sham interventions, you get a very clear group difference in the randomized clinical trial, and a very strong correlation between the achieved behavioral effect and the degree of modification of that altered EEG pattern. And we're now embarking studies to try to see how long can we sustain those benefits, how safe is it to do so, and how can one combine it with other interventions to get a more sustained effect. In addition to that, is we're thinking about the fact that this is just targeting one spatial temporal signature, one network linked to one set of symptoms. Oftentimes in cognition-related alterations of brain function, there is more than one symptom problem. For example, patients may have memory difficulties and executive dysfunction. If we can target more than one such dysfunction at once, doing a more complex multifocal montage, and we're embarked in an NIH-supported study now, looking at that and showing that the specificity of the intervention is really given by the targeted spatial temporal signature alteration and the induced change. So the idea is that it is possible to identify, despite the rather crude methodologies that we have in humans, patterns of altered spatial and temporal signatures that map onto symptoms and disabilities, and that by modulating them, we can reverse the alteration, improve the symptoms, and potentially might have a longer lasting, more sustained impact on the pathology itself. And that provides the promise of a personalized neuromodulation that needs to get a physiologic, individualized, measured signature of the impact of the stimulation to define the target in the intervention, but then allows for a home-based neuromodulation, which I think is an opportunity to start helping patients while learning fundamental insights in brain function. And I'll stop there, and thank you for the opportunity again.
Thank you, Alvaro. So this is very thought-provoking idea, and it resonates with some comments that I heard from our colleagues using fMRI, that only 30% of the variance is conserved across individuals. Essentially means that each of us has a different brain, and that it's hopeless to try to devise therapies or even diagnostics in the modern era without doing mapping of brain activity. So that means that essentially all the methods, including pharmacology, that we've used on patients in terms of neurological or mental disease has been are to be almost useless. No? So would that be?
Yeah, it's a good point, Rafa. So I think the point I would like to make is that it should be an experimental question. What I mean is that by not measuring, by not capturing the effects carefully, we lose the opportunity to actually find out the answer to the very question you're saying. It may well be, I can envision that it may well be that for some interventions, it's good enough to do sort of the same modulation in everyone. Yes, we're different, but there might be a commonality of effect that is crude enough in a way to translate into a benefit. So it may not be that you need to individualize intervention for absolutely everybody, but the promise would be that, or the hope would be that by doing the latter, by personalizing, the degree of efficacy, the lack of side effects, and the sustainability of the effects would be greater. So that's how I think about it. So I think we're making our life both easier and at the same time incredibly hard, because by using the stimulation without that level of precision, what we end up inducing is a murky water of uncertainty, whether what you deal with is individual variability, is variability of the effects, is the fact that some people don't respond. So I think at the very least, we should get careful assessments of the effects to be able to disentangle the answers, basically.
So there's two questions from Iñaki Ortego Isaza. Thank you for your great presentation. Most of the papers using repetitive TMS, they always explain the high frequency protocols as excitatory and low frequency as inhibitory. But as far as I understand, you say that this effect is patient dependent. And the second question is very related to the, I suppose that the only way to be sure what we want to do is to do it in a closed loop manner, monitoring brain activity. No?
Yeah, so thank you, Iñaki, great questions. The way I think about it is exactly the point you're making. We should not think of it as intrinsically excitatory or inhibitory. They are not. It's also not really patient dependent alone. It's more brain state dependent in addition to individual dependent. So I didn't get into it, but you can literally change within an individual the effect of the stimulation by engaging in a behavioral or another technology-based intervention task that shifts the state of activity in the brain. Therefore, your point of closed loop and monitoring becomes essential because effectively, by tuning the stimulation, by gating it to specific points, even on the EEG, you get a very differential effect. And so the promise of closed loop, I think, would allow at least to explore those questions. And I think there would be great value in that.
There's a question from Alia Benali. Could the effects in the NIBS be an effect of having no good controls?
Yeah, thank you for that question, Alia. I think your point is well taken that it is oftentimes challenging to define good controls, good sham controls for non-invasive brain stimulation, but it's not impossible. So, for example, for transcranial current stimulation, we develop a technique called ActiSham, in which what you end up doing is inducing the exact same amount of current in the individual as in the real condition, but placing electrodes such that the field ends up not penetrating into the brain. It creates, if anything, more current density in the skin. The perception of the subjects, even those who are experienced with the brain stimulation, is to be truly blinded because you feel the same sensations. But we can show through neurophysiology and particularly through careful modeling that nothing is being induced in the brain. So I think it is possible to create appropriate controls, but your point is very well taken. A lot of the published literature doesn't have good controls, and so therefore it is quite reasonable to question some of the things that have been published, including by me and others, and keep exploring the question in more detail now that we have the tools to do so.
Thank you, Alvaro. So with this, let's move to the next speaker, the keynote speaker. And Alvaro, please, if you could remain in the meeting, because we'll have a panel at the end after the keynote where all the speakers will participate. So, so it's a pleasure to introduce Adam Cohen. Adam is a professor of chemistry, chemical biology, and physics at Harvard. He's another polymath. In fact, he's one of the most original colleagues in neuroscience today. I can confess that to you. And the title of his talk is Molecular Tools for Mapping Memories. Adam, you have 40 minutes for your talk, and then we'll have a 5-minute Q&A with you, and then we'll roll it into the panel discussion.
Great. Thank you very much, Rafa, for the very kind introduction and for the invitation to join this interesting session and these discussions. And thanks to everybody who's online virtually for coming here. Is my screen coming through?
Yes, it's perfect.
Great. Okay. So I want you to take a minute and think about what you had for breakfast this morning. So there's some three-dimensional representation of that memory inside of your brain. And we'd really like to understand what is it about the patterns of activity during your breakfast, which caused some long-lasting changes in your brain. And then what is the three-dimensional representation? What were the patterns of synaptic plasticity and changes in gene expression, which give you the ability to recall something that happened over a timescale long compared to the basic timescale of spiking the basic events of neural activity. So this is a question I've become quite fascinated by. And one of the both challenges and interesting aspects of it is that this integrates dynamics across a wide range of scales of space and time. There are the fast spiking dynamics and even subcellular bioelectrical dynamics associated with neural activity and activity-dependent plasticity, which happen on the micron and microsecond to millisecond timescales. That drives changes in synaptic strength, which are still over very small scales, but can occur more slowly and can last for a long time. And ultimately, there can be changes in gene expression, which might occur over very broadly distributed circuits in the brain. There's clearly no tool which can measure all of these things. And so we've been developing a family of tools to try to get at different aspects of these dynamics with the hope that we can somehow then piece the parts together. And so today I'll tell you 3 vignettes about different tools for measuring these 3 different processes. And I'll focus mostly on the tool aspect and then give just a little flavor of some ways in which we've used these tools to start to learn some things. So let's start with the subcellular details of bioelectrical dynamics. So in a cartoon representation, a neuron is a device that takes synaptic inputs and it generates spikes. And those spikes go down the axon where they can trigger more neural activity. And they also propagate back up the dendritic tree where they can interact with synaptic inputs to drive plasticity. And so one question we'd like to explore is the integration rule. So what is the function, or it's really a set of functions, which relate different patterns of synaptic inputs, these could be excitatory, inhibitory, or modulatory, to the pattern of spikes. We know that the dendrites contain many nonlinear elements, and so there's some logic function or nonlinear computation which relates the inputs to the outputs. Then we'd also like to know how the inputs and the outputs interact together to change the input-output function of a neuron. That's what we call plasticity, or the learning rules. And so that's another set of things we'd like to understand. Now, many beautiful experiments over decades have characterized a zoo of different nonlinear excitations which can arise in different parts of a dendritic tree. So there's calcium spikes, sodium spikes, NMDA spikes, and so on. But what hasn't been clear, at least to me, is what's the relation between these different biophysical processes and the underlying information processing in the neuron. And so our goal is, our hope is that if we can map bioelectrical signals in the dendrites, we might try to understand something about how information processing emerges from the biophysics of the dendrites. So to start, we need a way to map bioelectrical signals, and our efforts in this area start here. This is the Dead Sea between Israel and Jordan. There are microorganisms which live in the water here, which produce proteins, which sit in the membrane. They absorb sunlight, and they pump protons across the membrane. That establishes a proton motive force, which these creatures use to power their metabolism. More than a decade ago, I had this notion that maybe we could somehow run these proteins in reverse. We could use them to convert changes in membrane voltage into an optical signal. And pretty much by luck, we discovered that these proteins are a little bit fluorescent. They can be excited by red light, and they emit in the near infrared, but they're only fluorescent when a proton is sitting on a particular spot inside the protein. Now, protons are charged, so if there's a change in the membrane voltage, that can push the proton onto the protein or pull it off and modulate the fluorescence. Okay? And so this turns out to work. Here's a HEC cell, so just a cell in culture expressing this protein straight out of the Dead Sea. And you can't see it, but there's a little patch pipette in the cell. And I'll show you a movie here where we're applying a ramp of voltage, and you can see the fluorescence go up and down as the voltage shifts. And it's a roughly linear response, and it's very fast enough to see action potentials. The challenge with this approach is that the fluorescence is very dim. It's about 100-fold dimmer than GFP. And so then we and other people subsequently have explored the idea that you could attach a different fluorescent protein, a conventional fluorescent protein, or dye, to these voltage sensors. Voltage-induced changes in the absorption spectrum of the opsin lead to quenching of the attached fluorophore, which then modulates its fluorescence, which is much brighter. And so here's an example from some of our early work with M-orange attached to this opsin. And as the voltage goes up and down, the M-orange and the opsin have oppositely directed voltage changes. What you pay here, the trade-off is that the sensitivity is a little bit lower because it's an indirect readout, but the brightness is much higher, and under some circumstances, that's a favorable trade-off. One of the charms of having a far red voltage indicator is that you can combine it with a blue light activated ion channel so that you can stimulate a cell with flashes of blue light and then record from it with red light and have an all optical interface to neural activity. And so here are some example recordings where the blue bars represent flashes of blue light to cultured neurons, and then the red is the fluorescence, and the black is a simultaneous patch recording for ground truth. And with these capabilities, it's now possible to record in a very wide field of view. These are cultured rat hippocampal neurons. There's about 700 neurons in this field of view. And this movie is slowed down about 50-fold from real time, but we can stimulate them in arbitrary patterns of space and time and record every spike and every subthreshold wiggle in all of the neurons in the culture. So it's a complete neuro-optical interface. All right. So now I'm going to tell you about how we used this to explore dendritic excitability. Our instrument uses two digital micromirror devices. These are basically video projectors. One to pattern the blue light for targeted optogenetic stimulation, and one that patterns orange light which we use to just illuminate the dendrites that we want to record from in order to enhance our signal to noise ratio. We also have a 2-photon microscope connected to the system for structural imaging. We'll use a light-gated ion channel for stimulating the cells, and we'll use a voltage indicator that was developed recently at Genelia Farm called Voltron 2 with a red dye that is charged into it so that we can do high-resolution voltage imaging. This is what the setup looks like when it's running, so it looks sort of like an atomic physics experiment. And now I'll show you some data. So this is a view of a CA1 pyramidal cell in an acute hippocampal slice from a mouse. And we'll target the blue light here just to the cell body. And if we look at the resulting fluorescence of our voltage indicator, we can see every time we flash the light at the cell body, we can see a spike there. But now part of the charm is that we can now map how these signals propagate through the dendritic tree. Here's an example where we'll stimulate the cell body. And this is a computed map where we fit the waveform of the fluorescence at each pixel in order to infer the timing of the wavefront passing that pixel. And then we make a new map where we sort of interpolate to very high time resolution to show how the wavefront propagates through the dendritic tree. And this is a movie where we're processing each pixel independently, so we don't impose any wave-like motion on the data. Any waves we see are really there in the raw data. This is at 40,000 frames a second. And you can see in detail how the action potential propagates through the dendritic tree and decays as you get further out away from the cell body. Okay, now things are going to start to get a little bit weird. So now we're going to start to get into some of the biophysics. We're going to continue to stimulate at the cell body, but we're going to give longer stimuli, which evoke trios of spikes at the cell body. And we're going to look out in the distal dendrites. And look at this. We see two different kinds of responses. Sometimes those 3 spikes at the cell body give a very stunted and low-pass filtered response out in the distal dendrite. But sometimes we get a single large event out in the distal dendrite. So this is clearly a nonlinear response. It happens sometimes, it doesn't happen sometimes. 3 spikes at the soma lead to 1 so-called dendritic spike out in the distal dendrites. And so now we'd like to map the spatial structures of these events and try to understand what's the relation between the pattern of inputs and whether or not we get these dendritic spikes and what their spatial footprint looks like. All right, so as a warm-up exercise, let's look at the sub-threshold voltages induced by stimuli in different parts of the cell. So these are, each row here is a different image of the same cell. And we're going to use our video projector to target the channel rhodopsin stimulation to different noodles here in the dendritic tree, and we'll see what happens. And if you look at the sub-threshold responses, your red is larger response, blue is smaller, you can see that those, perhaps not surprisingly, basically follow the pattern of the stimuli. So wherever we stimulate, that's where we get the largest subthreshold response, and then it decays with some length constant away from the stimulus zone. This is not terribly surprising. Now let's look at the patterns of the back-propagating action potentials, the ones that decay, as well as those dendritic spikes, the ones that don't decay, as a function of where the stimulus is. And before doing these experiments, I had expected that we might see some sort of channeling of those dendritic spikes, of the nonlinear events, into the dendrites that we stimulated, that somehow those dendrites would be sensitized and that would, there would be some information about where the input came from, which evoked a dendritic spike. But that's not what we saw. So here I'm showing the spatial footprints of the back-propagating action potentials and the dendritic spikes for each of these different stimulus locations. And you can see that to a reasonable approximation, neither of these two types of events depends really very much at all on where the stimulus is. It seems like all of the knowledge or the information about where the stimulus came from has gotten lost by the time we get a spike. And either we get a BAP or a dendritic spike, but those are stereotyped events. And so it looks like the cell really only generates two kinds of events, a BAP or a dendritic spike, independent of where the stimulus came from. That was a surprise to me. But we found that there is information about the stimulus location in the timing and probability of getting a dendritic spike. So here are histograms showing the number of events, either BAPs or dendritic spikes, as a function of time after the onset of the optogenetic stimulus. If we stimulate the soma, we almost always, but not exclusively, get BAPs which decay. Occasionally, on the third event, we get a dendritic spike. If we stimulate distally, we almost always get a dendritic spike on the first event. If we stimulate both distally and at the soma, we get two spikes which decay, and then the third one, 100% of the time, is a dendritic spike. Okay, so what's going on here? This is a somewhat strange pattern. One theme which emerged from this, when we looked at many, many cells, is that the probability of getting a dendritic spike on the first event was a rather steep sigmoidal function of the distance of the stimulation from the soma. So if you stimulate very distally, you're much more likely to get a dendritic spike on the first event, whereas if you stimulate just at the soma, you almost never get a dendritic spike on the first event. One can summarize all of these observations by a sort of rule, which is that the distal dendrites need to be depolarized for about 15 milliseconds for a backpropagating action potential to trigger a dendritic spike. So you can see in all of these cases, the dendritic spikes never come early, and either you need a couple of backpropagating action potentials to sort of invade the dendritic tree and to cause it to depolarize enough to support a dendritic spike. Or if you directly stimulate distally, then it takes about 15 milliseconds for that signal to propagate to the soma, where it triggers a spike, which then backpropagates and makes a dendritic spike. Okay. So, and we'll come back to the biophysics of what's driving this in a minute, but this is an observation. One thing we never observed in these experiments is local generation of dendritic spikes in the stimulated dendrites. We always saw that the event started as a spike at the soma and backpropagated. And so there's a question which we'll come to later about why we weren't seeing local events being generated. Now, if we deliver a sustained stimulus, things get even weirder. Here, we're stimulating this noodle on the cell, and the soma spikes, gives a burst of spikes, and then keeps spiking. Out in the distal dendrites, we see that we get a failure, and then two dendritic spikes, and then a series of failures again. And this is a very reliable phenomenon. So for sustained stimuli, we get a window of dendritic spikes, which lasts from about 15 or 20 milliseconds after stimulus onset to about 80 milliseconds and then shuts. Okay, and so you need some time for the dendrites to depolarize enough to start spiking, but then after a while they seem to get tired and they stop spiking again. Okay, so I presented a series of phenomena, and now the question is, what's the biophysical basis of this? Can we relate this to something about the ion channels of the cell? And we've done some numerical modeling of these processes, and I think I have a relatively simple, intuitive explanation of what's going on. There are two types of ion channels that are the key players in this. There are the so-called A-type potassium channels, and then there's the dendritic sodium channels. These A-type potassium channels normally provide a strong shunt which suppresses backpropagation. But these channels have the property that if you depolarize a little bit, they shut, they inactivate, which is then permissive of backpropagation. It increases the length constant for the signals to propagate. The sodium channels in the dendrites can drive these action potentials, the dendritic spikes, but there's barely enough of them to do that. And the dendritic sodium channels can go into what's called a slow inactivated state, where after they spike a few times, they get tired and they're not able to drive spikes anymore until they're given some rest to recover. And so you have a competition between the inactivation of the A-type potassium channels, which opens this window for dendritic spike generation, and the inactivation of the sodium channels, which closes this window. And because those two things happen with slightly different time constants, you end up with essentially a high-pass filter on the spike rate. So when there's an increase in spike rate, then that opens this window for backpropagation and dendritic spikes. At any constant spike rate, you don't get these dendritic spikes because the sodium channels inactivate. And this is what you might actually want for a backpropagation rule, because you want to know not just is the neuron firing a lot, but has some input driven an increase in the firing rate. And the dendrites act as a spike rate accelerometer, permitting that to happen. So we've done these simulations, and they pretty closely reproduce our observations. So here's some simulated data showing that you get this failure, success, failure motif, and here's the data. A charm of the simulations is you can look under the hood and see what the different ion channels are doing. And what we see is that the potassium channels inactivate first, that opens the window. The sodium channels inactivate more slowly, and that closes the window. Okay. Another observation we made experimentally is that if we stimulated the cell broadly, we would get a period doubling bifurcation, where we would alternate dendritic spikes and failures back and forth, back and forth. And that was sort of a surprise to us. So you see the period out in the dendrites is twice the period at the soma. When we simulated in our model broad optogenetic simulation, we got the same phenomenon. So again, here again, you see this period doubling bifurcation followed by a series of failures. And so this is encouraging to us that this model really is capturing the full diversity of the phenomena that we see in the experiments. Intuitively, what's going on here is that under the broad stimulation, the absolute refractory period of the dendrites becomes longer than the absolute refractory period of the soma. And so after one dendritic spike, the next spike from the soma comes before the dendrites have recovered, so you get a failure. And then after that, the dendrites have recovered, so then the next one gives you a success, and you alternate back and forth. And we've actually seen both in experiments and simulations that if you drive hard enough, you can make the dendrites go chaotic, where you have pretty regular spiking at the soma, but you have a chaotic pattern of backpropagation into the dendrites. Okay, but with all of these observations, we still observed that the dendrites looked quite stiff bioelectrically. And what I mean by that is we either saw dendritic spikes, which were sort of global, or we didn't. We did not see localized events on different dendritic branches. And so this then raises a mystery, which is, how does the cell know which synapses to potentiate during a memory? Clearly, that information is not in the backpropagating action potential or the dendritic spikes, because those seem to have no memory of where the inputs came from. So those do not contain the memory trace. To address this question, we explored the role of one other ion channel, which we haven't mentioned so far, which is the NMDA receptor. When we do optogenetic stimulation to a cell, that is opening ion channels in the membrane, but it's not actually a synaptic input. There's no glutamate involved. And so none of the synaptic receptors are getting activated. For real synaptic inputs, we have glutamate, which can activate these NMDA receptors. And these NMDA receptors are just a miraculous molecular machine which act as a cross-correlator of glutamate inputs and bioelectrical signals. And what I mean by that is that for these receptors to open, you require both binding of glutamate and an electrical depolarization to remove this magnesium block. And so these are the things that you need in order to, they're necessary for learning, and they're molecular cross-correlators of presynaptic and postsynaptic activity. So to explore the role of the NMDA receptors, we then replaced some of the optogenetic stimulation by electric field stimulation with an electrode, which we use to stimulate the axons which pass by the dendritic tree. So now we can deliver real synaptic inputs to our cells. Here's what we saw. If we deliver just an optogenetic stimulus to the soma, and we map the voltage throughout the cell, okay, so we get 3 spikes here at the soma, and as before, the first 2 fail, and then the third one makes a dendritic spike in this example. This is what we saw before. If we just deliver an electric field stimulation, we get a depolarization, which in this case triggers a single spike at the soma. If we now pair the electric field stimulation and the optogenetic stimulus, now the cell goes nuts. And you can see you get this very sustained plateau potential out in the dendrites and this complex spike or burst at the soma. And you can see this event lasts well past the end of either of our stimuli, okay? And so our picture is that when the electric field stimulation primes the NMDA receptor with glutamate, and that then coincides with a dendritic sodium spike, that sodium spike is enough to activate the NMDA receptors, which then pass an inward current, which then drives the soma to spike more, and then you get this sort of ping pong of these waves between the dendrites and the soma, which drives the sustained plateau and this NMDA spike, which lasts until the NMDA receptors finally shut. So we then explored how this phenomenon depends on the relative timing of the optical and electrical stimuli. And not surprisingly, they have to overlap in time to get these nonlinear events. One neat aspect of this is that we only get these nonlinear NMDA spikes if we have 3 or more back-propagating action potentials. A single event, a single spike at the soma, is not enough to trigger one of these NMDA spikes. And this tells us that it's almost certainly the case that you need the dendritic sodium spikes to be the match that lights the fire, which eventually leads to the NMDA spikes. Just back-propagating action potentials by themselves are not enough to do that. And you can almost see that here. If you look at the details of the voltage, you can see that the first 2 action potentials at the soma here don't really get the event started. And then it's the third one which leads to this dendritic spike, which then gets this large NMDA spike going, okay? So we think we've sort of dissected here something about what it takes to get one of these NMDA spikes. These NMDA spikes are believed to be necessary for synaptic plasticity. And so here we have a plasticity rule, which is that you need a burst of spikes from the soma in order to overlap with a synaptic input in order to trigger one of these events. We've done some pharmacology to explore which channels are necessary for this, blocking the sodium channels with just a little sprinkling of TTX so that the soma keeps spiking, but the dendrites become non-excitable, completely abrogates these events. Blocking voltage-gated calcium channels also abrogates the events, and blocking NMDA receptors diminishes their amplitude. And so here's sort of my cartoon picture of what's going on. We need the voltage-gated sodium channels to start the fire. You need to get a dendritic spike. That probably triggers the voltage-gated calcium channels to then drive a more sustained event, which then is enough to sustain the depolarization to get an NMDA spike. And so here's a cartoon summary of everything I've said. If you look at the pure electrophysiology of the dendrites without any synaptic inputs, we see that the dendrites act as a spike rate accelerometer. So if you have an increase in spike rate at the soma, you get a transient window for dendritic spikes. If those happen to overlap with an NMDA, with a glutamate input, then you can trigger the NMDA spikes. And so the question of where does the memory trace live is answered in this model, that it doesn't, it's split between the voltage and the glutamate signal. The voltage signal carries very little spatial information, but has excellent timing information. And so it is a broadcast signal which tells all of the dendrites about what the spiking is doing at the soma, if you have an acceleration in the spike rate. The NMDA receptors have relatively low time resolution because they activate slowly and they hang on to the glutamate for a while. But of course, they have excellent spatial resolution. They tell you exactly which synapse got the glutamate inputs. And it's the product of those two things which activates the NMDA receptors, which is the trigger for plasticity. And so here's my cartoon of what's going on. Okay, so far we've talked about the fine-grained details of dendritic excitability, which we think underlie the initiation of synaptic plasticity. Now I want to zoom out and ask about what are the actual patterns of synaptic plasticity associated with a memory formation event. And I'm going to talk about some tools which are cousins of some of the tools that we heard about this morning for mapping when there are new AMPA receptors on the surface of a cell. So we think that synaptic plasticity involves, among many other things, exocytosis of new AMPA receptors in the dendritic spine, which can then increase synaptic strength. To probe the delivery of new AMPA receptors to the surface, we developed the following construct. It's a fusion of a halotag receptor with a glutamate receptor. So this halotag is a non-fluorescent protein, which can be charged with a fluorescent dye. Some of the dye ligands for this receptor do not cross the cell membrane, and we're only going to use cell impermeable dyes. So the idea is that we'll express this construct in neurons. We'll deliver a first dye, which will saturate all of the AMPA receptors on the cell surface, but not the ones inside the cell. We'll then do something to the animal, make it learn something somehow, and then we'll inject a second dye. And any new AMPA receptors on the cell surface will take up the second dye. And so then we'll image the distribution of the first and second dye, and that will tell us where there has been AMPA receptor exocytosis, which we think will be a model for plasticity. And so here's a picture of a neuron, again, in CA1 expressing these constructs. Here we've put in a just broad membrane label in green, and then labeled the halotag GluA1 in red. And you can see it's nicely localized to the synapses, as we would hope. And now here's our experimental protocol. We're going to express the stuff in the mouse brain by in utero electroporation. And then after the mouse has grown up, we will first inject one dye, and then another dye, and then we'll shock the mouse's foot. So we'll do contextual fear conditioning. And then we'll look at where are the synapses which are displaying the second dye. So that's an ex vivo measurement after the fact. And here's a map showing the distribution of the potentiated synapses in CA1 pyramidal cells. Here, we've overlaid 19 different neurons all on top of each other. So that's why it looks like such a mess of neurites. But the point of doing that is so that you can sort of see by eye what the distribution of plasticity is. And it was quite striking that the plasticity was primarily localized near the soma in both the basal and proximal apical dendrites. This is consistent with the current understanding of how plasticity works in CA1 pyramidal cells, that distal inputs provide an instructive pathway of whether or not plasticity should occur, but the plasticity is primarily in the CA3 to CA1 synapses, which are closer to the soma. This cartoon is also consistent with the picture that I presented of the dendritic excitability, where the distal inputs are what are needed in order to trigger these NMDA spikes, which we think are the trigger for synaptic plasticity. And so we think we're starting to develop a biophysical understanding of this cartoon. Now, historically, people have used many different surrogate markers of plasticity in the brain. Perhaps, well, one common one is to look at CFOS, which is an immediate early gene, which is a marker of high neural activity. And people associate this with so-called engram cells, cells which are somehow involved in formation of a memory. And so one natural question is, what's the relation between CFOS expression and synaptic plasticity, which, if you study synapses, you think is the basis of memory? Yeah, these are supposed to somehow tell us about the same thing, but the actual relation between these two things had not previously been measured. And so we looked at a bunch of neurons where we counted what fraction of the spines were potentiated. And we also stained their cell bodies, their nuclei, for CFOS. And we looked at how much CFOS expression there was. And to my great surprise, there was a quite strong correlation between these two parameters. And so this, I think, perhaps provides some basis for using CFOS as a proxy for synaptic plasticity. One of the questions in studies that use CFOS to mark engram cells is, what is the actual physiological change that the CFOS is reporting on? And this suggests that at least one such change is the amount of synaptic plasticity. All right. So now I want to zoom out even more. And instead of looking at plasticity of individual synapses, I want to ask about brain-wide changes in gene expression. So we think that the changes associated with, say, a memory of your breakfast might be broadly distributed across the brain. And so the question is, how can you map such changes when there's no way we can image the entire brain, at least of a mouse, inside the live animal? And so we'd like something which can record for very long times across the whole brain, across multiple modalities. And so our approach to that is inspired by the way ecologists can record climate history by looking at tree rings. So if you cut across a tree, you can learn about climate events from hundreds of years ago by looking at these tree rings. And you can do that throughout a forest, and you can map climate histories. We wanted to do a similar thing inside of neurons. So to do that, we need three ingredients. We need some linearly growing scaffold, which will grow over time. We need some timestamps akin to the seasonal bands on the tree rings, which mark defined increments of time. And then we need some physiological marks, something that tells us about what the neuron was doing at each of those times. We stumbled across a protein called PAC4, which, if you express it in a cell, spontaneously assembles into these rod-like fibers. The crystal structure of these fibers shows that they, in cross-section, have a hexagonal lattice, and along their length, they have these pores, which are about 8 nanometers diameter, which run the length of the fiber. Those pores are big enough that the protein can be decorated with different payloads, which will incorporate into the pores. And so if you decorate with GFP, you get green fluorescent fibers. If you decorate with a halo tag receptor, you can then load up these fibers with different colored dyes and give them the corresponding color. So here's our scheme for using this to try to record from neurons. There will be three ingredients. We'll express the PAC4 so that it forms fibers inside the cell. We'll then also express under a constitutive promoter the halo tag fusion, and we'll periodically inject different colors of dye. Each dye will, the transitions from one dye to the next will mark the times when we switched the dye. And then we'll have, under an activity-dependent promoter, a GFP fusion, a third color, and we hope we'll get green stripes in these fibers when the neuron is active. And the location of those green stripes relative to the color bands will tell us about the timing of the neural activity. here's what these fibers look like. Here we're expressing them in hex cells, and they grow from both ends. And so you get 2 copies of your color pattern on the 2 ends of the fiber. So here we stain with red, blue, green, and back to red. And you can see that on both sides of the cell, of the fiber. Now, one attribute of these fibers you can see here is that they're quite stiff, and they actually get quite a bit bigger than the cell. And so one challenge, which has prevented us from doing this in vivo so far, is that if you did this in the brain of a mouse, you would sort of shish kebab many of the cells in the brain, which you probably don't want to do. But nonetheless, we decided to try doing this in cultured neurons. And so here's the protocol. We express these constructs in the neurons. And then at first, we added a yellow dye. At t equals 0, we added a red dye. And then at either 3, 6, or 9 hours, we added a drug which goosed up the cells, which triggered CFOS expression. And then at 12 hours, we switched back to the yellow dye. And here are some images of the fibers. I've separated out the green channel here. And you can see that the onset of the green fluorescence in the fibers reflects the time of the drug addition when we measure the green position relative to the yellow to red and red to yellow transition. And if you look at many of these fibers across many of the cells, you can see that there's a nice linear relationship where we can infer the timing that the neuron was activated with a precision of about 30 minutes over a 12-hour baseline. So this isn't cultured neurons. This is really still a technology in development. It's not ready for prime time application in live animals yet. We're exploring a variety of different scaffolds with the hope of finding something that's more flexible so it doesn't disrupt the neurons. We're exploring other ways of time tagging the fibers so that instead of having to inject these dyes, maybe we can just use flashes of light. And we're exploring a variety of different modalities one might record. So not just gene expression, but also looking at things like calcium signals or kinase activity. And our hope is that one could express these tools broadly in the brain, timestamp them, record in an animal doing some set of learning tasks and recall, and then take out the brain and do very wide area imaging and look at the patterns of neural activity across time and across the whole brain. That's still a hope for the future. So to summarize, I've told you about a series of tools for mapping neural dynamics ranging from microseconds and milliseconds and microns all the way out to centimeters and hours or days. And I hope that with these tools, we'll start to be able to put together mechanistic understanding of memory encodings. I want to end by just commenting on where these tools come from. The voltage indicator comes from here in the Dead Sea. The channelodopsin comes from an alga from a pond in England. We use proteins derived from corals and jellyfish. And it's just amazing to me that we can take these molecular nanomachines and staple them together and come up with these new functions. And we can only do this because ecologists and botanists have been describing these exotica for many decades. And so it's very important both to conserve and to describe the vast diversity of the natural world. These are the people who did the work. We've had many fantastic collaborators on the work that I told you about today. And of course, all the credit for the data and the work goes to the students in my lab who did the work. These people pay the bills. This is my lab. I'm always looking for people to join us. So if you're interested in these kinds of questions, please get in touch. And I'm happy to take questions. Thank you.
Thank you, Adam. I want to add to why don't we just turn this into an open discussion with all the other speakers so that they can also ask you questions. So if you could just turn on your camera, and we have just about half an hour to talk about this session. And there's actually a question for Adam from Divya Chander. What is the role of spatial configuration of the dendrite in shaping input-output functions at the level of the synapse and at the level of the entire neuron?
it's a good question, and the answer probably will be different for different cell types and will depend on a lot of details that we have not yet explored. The main effect that we've observed is that as you get further from the soma, of course, the voltage becomes more decoupled from the soma. But at least in these CA1 pyramidal cells, you can do a pretty good job describing the dynamics by just two compartments. So we're not talking about voltage in the spines themselves. That's, of course, a whole other world, which Rafa is an expert in. But at the level of the dendrites, they either spike or they don't. And that seems to be a broadly distributed phenomenon. And so, I think exploring the role of more detailed aspects of the morphology is something still for the future.
Adam, another question from Divya. What about noise in the synaptic input? Does noise improve or hurt the temporal and spatial resolution?
That's also a great question. And again, the short answer is I don't know. I think it's a good question, and I think it's something that would need to be explored in more detail.
Question from Karim Owais. Adam, great talk. Have you tried glutamate uncaging to selectively activate specific synapses of the dendrites as opposed to electrical stimulation?
Okay, so that's a very interesting question. And we're in the process of building the uncaging apparatus right now, specifically to do that. And so the short answer is no, we haven't done it. The reason I think that's an interesting thing to explore is that when we did our electric field simulation, that was a very blunt hammer. We were probably activating synaptic inputs across the dendritic tree. And so perhaps it's not surprising that the events that we saw were global. One could imagine that if you activate NMDA receptors specifically on a single dendritic branch, then that branch becomes more excitable than the other branches. And under those circumstances, we might start to see branch-specific dendritic excitations, which so far we haven't seen. And so it would be very interesting to explore what are the conditions for getting branch-specific excitations. Of course, ultimately, everything we do in SLICE is possibly different from what one might see in vivo. The overall neuromodulatory tone, the role of inhibitory inputs, and the overall level of excitatory inputs can change things. These neurons are so nonlinear that we can't really know what's happening in vivo until we look in vivo.
Adam, a quick question. Why do you choose Voltron?
So we have tried many different indicators, and we found that Voltron 2, so these dendrites are really small. And because of that, and looking in tissue, we have to worry about the background from all of the stuff that's out of focus. And so brightness was a key parameter there. And Voltron 2 is very bright. It's also reasonably sensitive and quite fast. One of the challenges in getting this to work was finding a dye which is far enough to the red to be paired with channel adoption without optical crosstalk, but still gives a good voltage response and is bioavailable in vivo. And that actually took quite a bit of exploration to identify the right dye there.
Why don't we open the discussion to all the speakers? And I have a burning question for Alvaro. You could take the next step and carry out a lot of the personalized neuromodulation in not in patients, but in normal people to enhance their mental and cognitive abilities. So what are your thoughts of that, about that? Has that happened, actually? Is that happening?
It's been working on me. No, I'm kidding. I mean, I think it is a burning question. It is a remarkably important ethical question, and unfortunately, it is happening. I mean, there are experiments being done, experiments having been published in humans with questions as open as, can we induce lasting plastic changes in humans? And you go like, wait. First of all, let's define what the heck you're doing. And second, how are you measuring what the long-term consequences of this may be? So I think there is a need to take the potential effect seriously and really carefully weigh what are questions that should be pursued in humans, what are questions that should be pursued first in animal models, what are things that should be pursued at all. One of the things we haven't done systematically enough, I think, in humans is to explore sort of models of the effects of the stimulation, brain models of the stimulation that could give us answers before actually experimentally trying things out in humans. So I think it's a very hot debate in the field. I think it links to a lot of questions regarding neurotechnology, ethics, implications, and I hope that regulations are put in place that are not limiting of the exploration and innovation, but that preclude or curb temptation that can go into the wrong direction.
I understand that there is a vibrant field of makers that build their own magnetic stimulators, and they stimulate themselves every day even, and they call it microdosing. Are you aware of that, or what are your thoughts about all those people, or your recommendations to them? And some of them could be listening.
Yeah, so I am aware of them, and I'm on record. We wrote a letter, an open letter to the do-it-yourself community of NIPS, of non-invasive brain stimulator, trying to accomplish two things. One, make people aware of the many things we don't know and the potential downside of those things we don't know. It's tempting to bring a slew of optimism bias and say all the outcomes will be positive, and if they are negative, they won't happen to me. But it's a little unrealistic to approach things that way. And so we wanted to at least highlight some of the core unknowns. And the second is, I think people are going to continue to do it as they are doing it. And so we offered the opportunity to actually engage with that community in a way to not regulate, but to try to extract knowledge from their experiences, to actually leverage what they're doing. The latter has not really happened, and I hope that the former, that at least the awareness might at least inform the approaches of some. I think it's worth people being aware that it's led to significant complications, to the point that as a physician involved in brain stimulation, I've seen a number of patients, healthy individuals, who through their self-application of stimulation have developed problems ranging from burning themselves significantly because of the amount of current applied, to start to experience feelings or sensations that they feel are related to the stimulation and don't like anymore. So this notion of enhancement is tempting. There are companies developing it. There is a lot of do-it-yourself community. It's deceivingly simple to do these tools and to apply it. But there is a lot of unknown questions that are important to unpack.
Yeah.
Thank you.
Rafa, I have a question for Alvaro, which probably you had a million times coming from physicists like me. So you said something about it in the beginning of your talk, but what might be the physical mechanism making this magnetic field stimulation work? Because to me, nothing should work, but it does work. So something's happening.
It's a good way to put it. I think something is happening. I'm hoping that physicists like yourself, biologists like all of the other panelists, Rafa included, will provide fundamental insights. I think, frankly, one of the things that has been insufficiently leveraged is the fact that, as I was in very brief passing pointing out, these techniques are scalable to induce similar currents, if nothing else, in similar orientations in animal models, and getting real insights from the animal models, applying stimulation that is commensurable to what we do in humans, and gaining biological insights at cellular ensemble levels, is something that very little has been done with, which is in many ways surprising, right? I think it's a manifestation of both the desperation that as physicians we experience and as patients they experience, and the deceivingly simple thing, I apply stimulation somewhere and hey, something happens, this is cool, and not realizing the many degrees of freedom that are between those two facts, that you applied something and something happened, right? And so I think there is a need for a more systematic filling in the gaps. If we actually want these technologies to truly expand, we need to understand mechanistically a lot more of what they're doing.
There's also a lot of room for theory, I guess, right? Because I mean, the ideas are not that complicated. I am a theorist myself, doing physics, and so at the end of the day, it's just electromagnetic fields and some charges that you have there in ions, and there are a lot of things that could be, I don't know, treated. I mean, say no, or yes, or I don't really know what your face means, but you clearly have something to say. Maybe do you want to jump in or?
Yes, thanks. So I actually had a perhaps somewhat related question for Alvaro. It's a two-parter. So the first part is, can you say something about the spatial resolution and depth penetration of the current stimulation tools? And then the second part is, can you say something about your wish list? What level of depth penetration or change in spatial resolution do you think would have sort of the greatest impact, or what would you like to see the ability to do?
Those are great questions, Adam. So very briefly, the answer obviously depends on which tool we're talking. If we're talking transcranial magnetic stimulation, and you scale down the coil without getting into the development of what we call micro coils that Giorgio Bonmassar and others are developing. But if you don't go there, the standard available coils will have a spatial resolution of about the tip of a little finger, so somewhere in the neighborhood of few cubic millimeters of cortex, and the decay is very steep with the square of the distance, so that largely you get an effect that is centered at cortical layers. The challenge is that there is a lot of shown to be relevant orientation effects. You can think of it as really pickup coils, and so if you use DSI to get main vectors of orientation of the cortical voxels, you can align your coil and either maximally activate or not activate at all a given voxel is captured by Bolt imaging, for example. And so even though the penetrance may be whatever it is, if there are fiber bundles that take a sudden bend and therefore go from maximal to completely out of orientation, you may have a depolarization at a distance from that cortical aspect. The second thing worth mentioning is, as I said, there are development of microcoils. There is also the development of coils that have different planes of fields. And so in doing so, the decay is not quite as steep. They're often referred to as deep TMS. I personally don't like the term because it leads you to believe that they are selectively stimulating the depth, which is not the case. They're still stimulating more in the surface. They just reach deeper, to your point, and those coils have been shown to reach subcortical basal ganglia structures. So quite deep in terms of sufficient current field to depolarize there. If the question is about transcranial current stimulation, as I said there, you can use the temporal interference to have selectivity in the depth for real there. And the limitation there is the dispersion of the field. It's at the level of getting activation of the amygdala or accumbens, but much like we would like, the resolution is too poor to get subnuclei of the thalamus, for example, at least at this point. Again, orientation may be allowing us to do something. My wish to start there would be to be able to combine those stimulation techniques with other technologies, think nanoparticles or similar approaches to have greater selectivity in the effect. We can do a little bit of that by engaging neuronal populations in activity, changing their state compared to the rest of the population, and show state dependency, greater selectivity. But to be able to do that with a systematic way would be very nice. I mentioned the closed loop effect, just to add one more, it's again largely a wish thing, because we don't have a really good way to close those loops. And the final thing is to learn from all of you what are the best way to monitor the effects of the stimulation while stimulating is probably a critical component that we need.
I know Alvaro has to leave in a couple of minutes, but maybe Alvaro, you may have time for a question from an anonymous attendee. Dr. Pasqualeone, any role of non-invasive neuromodulation for treating migraines?
Well, it's one of the, thank you for that question. It is one of the indications that has been very heavily studied as an off-label application. And there is in fact a device that was FDA cleared for migraine treatment as an abortive treatment of migraines. Interestingly, it's an interesting technology because it was developed as a handheld, home-based self-stimulation device. And the notion is that you can self-administer a pulse at the right time of a spreading depression. And in an experimental model, you can show you can break the spreading depression. And so the hand-wavy thought was we can do the same in humans in vivo. And so here you go into the simulation. It works. The problem is that only a small number of migraine patients have consistent auras that they can time appropriately when to stimulate. And if you don't, then your stimulation doesn't really have sufficient effect. And the data that you prevent further attacks is to me unconvincing. So there is some suggestion of potential effect, but insufficient. Quite different for neuropathic pain, where the data for the effects of neuromodulation are quite strong, I think.
Wonderful. Thank you, Alvaro. I think we're going to let you off the hook. Thank you for your participation. Maybe we can focus questions on the other speakers. In fact, I have a question for Juliette. So the electrowetting lens is super cool. What do you think it's its niche? So besides, I mean, you're sort of developing it to be used for fiber-based imaging systems, but could it be also an alternative to spatial light modulators in optical systems? What do you think are the good things and the bad things about the electrowetting lenses?
Well, so that's a great question. So first of all, I guess I just want to say that, we've done numerous studies with our wavefront sensor and then also with imaging campaigns, and showed that the quality of them is very, very good and comparable to what you can get from fixed micro lenses. In some sense, the imaging application is the most demanding one. You can get away with a lot more, for example, in LiDAR. So the electrowetting lenses do well for applications that need small devices. They also do well in applications that need low power. Let's see, they're quite versatile and can operate over a wide wavelength range. I guess specifically with regards to spatial light modulators, because this is something I've thought quite a bit about. The spatial light modulators use a CMOS backplane, and then you essentially have to do a few other steps, but you essentially sandwich the liquid crystal between two plates. And so you're able to address with the CMOS addressing. And so the big issue with the electrowetting is that we actually need sidewalls to contain our liquids and to make individual pixels. I was debating, but didn't have time to show some of our latest work on individually addressable arrays, which is, I think, one of the next places that we want to go, because it will, really open the application space. But I guess what I feel, maybe it's just sad to say, is I don't think the electrowetting array is going to replace the spatial light modulator. We do use spatial light modulators quite a bit in my lab, and we're fully aware of the downsides. One of them being speed. And we have reason to believe, for some of our published work, that the electrowetting lenses can get to kilohertz. And indeed, we showed with our LiDAR demonstration, one of the reasons I showed that is that the 200 hertz operation is actually fast enough for you to be able to build up an image. So I guess what I would say is, like everything else, it's got pluses and minuses. Maybe the pluses are that you get a large range of tunability, they're good quality, they're low power, they work best at the miniature scale. If you make them too large, then you run into the effects of density mismatch on the liquids. And so you start having effects of gravity, which is pretty undesirable. So, we're not going to make, the next liquid, liquid large mirror for a telescope. That's really not in the cards. But I think they jive quite nicely with things at the nanoscale. And I think another point, which actually turned out to be quite important for being able to do these miniature microscopes, is that they're transmissive. And so that makes a really big difference in terms of being able to compact something versus the reflective geometry. And I think to summarize, maybe the downside is that we have to have individual walls to contain our liquids. We do have some work looking at multiple electrodes and seeing how far we can get there. Thank you.
So questions. Actually, there's a question for Adam, a couple of questions from the audience. Other than the high amount of glutamate to increase the voltage, are there other potentials available that will transmit the same output?
Okay, if I understand the intent of the question correctly, perhaps it's a question about signals other than glutamate as ways of affecting dendritic excitability. And if that's the question, I think the answer is yes, there's quite interesting possibilities. So, we all have the experience that experiences that are somehow emotionally arousing are more memorable than things that are not. And so presumably neuromodulators play a very important role in gating plasticity. But the way, the details of how they actually do that is not clear. And so one thing which I think is very interesting to explore is how modulatory signaling pathways might affect dendritic excitability and these events that we're observing. And one could look at that either by delivering neuromodulators or by going inside the dendrites and perturbing things like cyclic AMP or IP3 and seeing what that does to the dendritic excitability. And that's an area where I think there's a lot of interesting possibilities to explore.
Thank you. I have a question for Lisa. So Lisa, you are your design is bio-inspired, but could you calculate ab initio the material? I work with the physicists like Itzel designing nanoparticles, where they can essentially they can design whatever they want to achieve. Could you do something like that also with metasurfaces? Forget about the butterfly, designing something that doesn't exist in nature, but could be even more powerful. Huh?
Oh, for sure. So that's a great point. We can definitely do ab initio calculations. I know Itzel's an expert in this as well. There's a lot of work on inverse design algorithms, just coming up with an absolutely randomized or random seeming shape that's optimized for a certain application. So in our case, we're kind of thinking of a more hybrid approach where we would use the nature-derived structure as an inspiration. So basically as a starting point in a sense. So we analyze SEM images of the structure and try to identify the key photonic features that give rise to this effect. But then we also think how we can surpass the effect with our key performance parameters. So basically the nature-derived system gives us a starting point because it works surprisingly well. But then we try to leverage that with our optimization approach to even surpass the performance that we see in nature.
Wonderful. I think both Itzel and Julie have their hands up.
Happy. My comment is very stupid that we talk so much about neuroscience that maybe I didn't tell you about many other things that we do in our lab, but we devote a lot of time to calculating these sort of structures. We design them using machine learning as well, so that they do what we want them to do. And it's an entire field of research, and yeah, I mean, it's a lot of fun. So one day I can tell you about it.
Wonderful. Juliet, I think you have your hands up. Yeah.
Yeah, so I had a question for Lisa. I was wondering how much tissue you can propagate through before you lose the signal, and then also whether there are various types of tissue that are easier to interrogate with your method. For example, OCT works really well on the eyes, but it's been more difficult to implement in other places.
I think we lost Lisa.
I think she's frozen.
Yeah. Or she got shocked by your question.
Hello.
Okay,
I think
you're right.
My connection. Julie, I heard the first part of your question.
So I guess I was wondering how far you can propagate through tissue, and then secondly, whether there are various types of tissue that are kind of more suitable for your technique. For example, OCT works very well on the eyes, but it's been a lot more difficult to implement in more scattering samples.
Perfect. Thanks so much for repeating that. I lost the connection briefly, but I'm back now. So yeah, that's a really good point. So we can look at, so of course, the thicker the tissue is, you have a propagation limitations for light. So in a first option, we can do several experiments with histological tissue samples that are optically thin, so around 5 to 10 micron in thickness, and there you don't have any issues with depolarization and so on. So that works very well. We have some really nice proof of concept experiments there. But of course, we can also look at optically thick samples. So the nanofibers I showed as a proof of concept, those are opaque. So there we actually just look at the back reflected light scattered off of the surface from the tissue. So we don't have a very deep penetration depth, of course, but we can still learn something about the structural properties at the surface. But in order to address the question of propagation depth, we're working on a separate project where we're trying to merge the physics of light and sounds. We're trying to generate patterned sound wavefronts that can propagate through the tissue at deeper penetration depth. So it's a whole other research direction. The materials physics of that system is actually really interesting and poses some challenges on its own. So that's been a really fun field to get into. But the longer term goal of that project is to really use the, combine the high resolution of light and then the high penetration depth of sound to address some of these challenges. And then to your question of particular tissues that are useful for this experiment. So our system, the one I presented today, the idea there is that we visualize the fibrous properties of tissue. So, certain tissues that have high collagen content are well suited, or certain diseases where changes in the fibrous structure is indicative are useful. So, for instance, scarring of the tissue is something where you would have a high collagen buildup. That's something we could visualize. If you were talking about OCT before, I know that's an issue, for instance, that one could look at there. But there are all kinds of fibrotic diseases across different organs where this technique could be applied. So because we're really looking at the biophysical properties of the tissue, just the fibrous tissue microstructure, it can actually be translated across different tissues and different diseases. So that's actually one of the strengths of our technology.
Lisa, there's a question from the audience, a little bit from left field. How can your technologies be adapted to things like neuromorphic computing and improved efficiency?
So I guess the field generally of nanophotonics, and I see Eitzel is nodding. I know he also knows a lot about this. There is quite a bit of work using these systems for neuromorphic computing. So my lab currently isn't working too much on that, but there's some really beautiful work from Nader and Geta and others, for example, that use these nanostructures known as metasurfaces to really solve equations themselves. So they can really be designed with some of these machine learning inverse design approaches to compute very complex mathematical operations with light, which is actually quite an impressive feat. So it's a very interesting and active field of research right now, absolutely.
If I can say a word or two, it's a field of research in itself. If anyone is interested, let me use this opportunity to make a little bit of publicity. We're organizing a conference in San Sebastian in person this time in September called Artificial Intelligence Photonics. And we're going to treat all these topics with some of the biggest experts in the world on this sort of thing. So if anyone is interested, ask Google Artificial Intelligence Photonics and it will pop up.
Thank you, Eitzel. I think the next time we do NanoNeuro2024, you need to speak about all the mischief that you're doing to meta surfaces with all your theory calculations. Yeah. So,
the keywords are similar, but the topics are very different. No neurons in no neurons.
Not yet.
Not yet. That's it.
So we're going to have to wrap up. We've been at it since 8 in the morning in the US or 2 p.m. in Europe. Maybe I myself just wanted to thank all the speakers. I think we had another phenomenal panel today. And of course, all the attendees for joining and putting up with the glitch that we had earlier on the Zoom. And I also wanted to thank and acknowledge the support that we got from the Chen Family Foundation, without which we wouldn't be able to have done this. And also I wanted to thank Lax Steinberg. Lax, could you turn on your camera? Lax has been the person behind the organization of all this conference that started a few months ago. So thank you, Lax. Also, without you, we wouldn't be here today. And then maybe the last thing is just to pass the mic to Eitzel so that he can say his last words to close the meeting.
Yeah, sure thing. So let me repeat your words. Thank you to every single speaker, to all the attendees that made an effort all around the world to stay up very late and to wake up very early. And also I would like to thank Luz, our person from the IT service in San Sebastian, who has been super helpful today, even if we had some problems in the way. And one last thing, I would like to announce together with Rafa that we have that we will soon have a couple of positions available in San Sebastian for a tenure track scientist and also for a more senior scientist in these topics of nano neurosciences. And so if anyone in the audience is interested in these positions, send us an email and we will send you more information. And that's pretty much it. So it's been another year. It's the 4th edition, the one that we are closing right now. And we are already thinking about the 5th. So hope to see you there. And thanks again for all the attention.
Right. Thank you, everyone.
Thank you so much.
Bye.
Bye.
Yeah, thank you. It was a real honor.